Good morning, everybody. It's really nice to see you all after what I hope has been a busy and productive summer. One of the things we mentioned early on when we had the first orientation was how important we think these kind of research opportunities are, regardless of where you are in your education. And I'm hopeful today that we're going to hear great things from all of you about what you've learned over the course of this summer and how you're going to take this forward, not just for the near term, but as you think about the future as well. So on behalf of the foundation, we're happy to have you here and excited about hearing about what you've done. So with that, I'm going to turn it back to Helena and get us on schedule for the day.
Okay, great. Thank you, John. Hi, I'm Helena Andraco. I'm administrator of the Hudson River Foundation, and I'm glad that you all could join us today. So I'd like to begin by introducing everyone that you see on your screen, and I'll begin with the Polgar Committee, who aside from myself is Dr. David Yazzo from Great Ecology and Sarah Fernald from the Hudson River National Estuarine Research Reserve. We have our 7 2021 Polgar Fellows, Nicole Steinbach, Royal McMahon Ward, Emma Butzler, Luana Chokai, Maximilian Brown, Ashley Smith-Krudener, and Beryl Khan. We also have some of their advisors here today, Dr. Zion Klaus from Marist College, Valerie Luzadis from SUNY ESF, Mark Botten from Fordham University, Allison Fitzgerald from New Jersey City University, Gregory O'Mullen from Queens College CUNY, and Chester Zarnock from Baruch College CUNY. We also have Emma Blackford here today, who is going to help us with technical issues. I'd like to go over the order of the day. We'll start with short presentations, giving some background on the Hudson River Foundation and Herner, our partner in the Polgar, by Dr. Yazzo and Sarah Fernald. We'll then hear from our fellows. Each fellow will have 20 minutes to present their work, plus 10 minutes for questions. Members of the audience, please write your questions in the chat. Fellows and advisors can raise their physical or virtual hands. And if you see that we've missed you, just call it out. It's a very long day, so we have to stay on schedule. So I apologize in advance if we don't get to your questions. We'll take 2 short breaks. And please remember when not speaking to mute yourself. Now I'll turn the program over to Dr. Yazzo. Dave, take it away.
Okay, thanks. Emma, are you going to pull up the presentation? There we go. Okay, I'm not going to actually talk about the history of the Hudson River Foundation, but I'm going to talk about the Polgar Fellowship Program, whose history is nearly as long as the Foundation's. Next slide, please. The Polgar Fellowship Program was initiated in 1985. Initially, the program was restricted to projects only within the Hudson River National Estuarine Research Reserve sites. And at that time, the reserve was new. It was started in 1984. The Hudson River component was, and so if you're not familiar with the reserve, the 4 component sites. Oh, back up. I think it's it jumped 2 slides, Emma. Okay, so the 4 component sites of the reserve are Piermont Marsh, Iona Island, Tivoli Bays, and Stockport Flats. However, since 1988, the program was expanded when the foundation took over underwriting the entire program, and it included projects within the entire estuary. In fact, since then, we've expanded the scope of the work to even include the non-tidal Hudson River, so it's really the entire watershed. However, many projects do still take place within the research reserve system because of all the opportunities provided there, which Sarah is going to talk about shortly. So 2021 is the 37th cohort. We've been doing this for 37 years. We've funded 291 fellows to date, awarded over $1.2 million in stipends. Next slide, please. So who was Tibor Polgar? Well, he certainly had an interesting life. He was the first chair of the Hudson River Science Panel, again, coming on board very shortly after the formation of the foundation in early 1980s. He had an adverse early life. He was born during World War II in a Nazi war camp in his native Hungary. He managed to emigrate with his family to New York City at the age of 15, where he attended high school. And then he went on to Amherst College to earn his BS. And then he went to the University of Rhode Island, where he got his master's and PhD in marine science. He then went on and joined the Martin Marietta Corporation and became the technical and scientific director of their environmental systems division. And as I said, also became the first chair of the Hudson River Science Panel. Martin Marietta is still in business, but they're known as Versar now. So if you've ever heard of the company Versar, that's actually the same company. Unfortunately, Tibor died at a very early age, right at the peak of his career at the age of 41, right about the time the Polgar Fellowship was getting underway. So of course it made obvious sense to name the fellowship program after him. So that's the story of Tibor. Next slide, please. So here's how the program works. We solicit research proposals. They are peer reviewed by the entire Polgar Fellowship Committee, me, Sarah, and Helena, but we also bring in at least 2 external reviewers for each submittal. So they are rigorously reviewed. It's a very competitive process. The duration of the research it's a summer program. It's 3 months, although most of the projects do tend to continue on as part of a larger effort, whether it's an undergraduate or a master's thesis, a dissertation, for example. So, even though we're presenting now on sort of the 3-month point, we do understand these are actually larger, more comprehensive projects than that. The advisors have a very major role in this process. They supervise all aspects of the research, they edit the final reports, and they participate in the orientation meeting and this final presentation meeting. The stipends are $5,000 to the fellows, and the advisors get $500, and each student gets a modest research budget as well. Next slide, please. And then it culminates in this event, the final presentations. Historically, we've been hosted all the years at the Carey Institute of Ecosystem Studies in Millbrook, New York. Unfortunately, starting last year, of course, continuing into this year, we couldn't do it because of the pandemic, but we certainly hope that eventually we'll get back to the Carey Institute. They're tremendous hosts, and it's a great event. And then, of course, the fellows publish a report. So we rigorously review the final reports, make sure they're consistent and technically sound, and we publish them in this bound volume, which the foundation distributes to libraries and institutions and makes it available on their website. But it is great literature, so we do encourage the fellows to publish their work in the peer-reviewed literature, and many, many fellows over the years have published. There's a tremendous body of primary literature that has come about from the Polgar Fellowship Program. And that's it. I'm going to turn it over to Sarah now.
Okay, thank you, Dave. Let me just bring up my presentation. All right, can everyone see that? I'm Sarah Fernald. I'm the research coordinator for the Hudson River National Estuarine Research Reserve. So the HRNR, as we like to call ourselves, is one of 29 research reserves across the country. We're all funded through NOAA with a shared mission to promote stewardship of the nation's estuaries through science and education using a system of protected areas. And like Dave mentioned, we have 4 component sites throughout the estuary, from south to north, Piermont Marsh, which is just south of the Mario Cuomo Bridge, Iona Island, which is in Bear Mountain State Park, Tivoli Bays is adjacent to Bard College, and Stockport Flats is just north of the city of Hudson. And pictured there is our headquarters at Norrie Point. Like I said, we are funded through NOAA, and that is delivered in partnership with the New York State Department of Environmental Conservation. We were designated in 1982, and across all 4 sites, we have 5,000 protected acres of freshwater tidal wetland. So within the reserve system, we have several different what we call sectors and different roles which we facilitate. I oversee the and monitoring program. I'll talk a little bit more about that in a minute. But we also have an extensive education and outreach led by Chris Bowser. They do a lot of work on eels and the tributaries and give some outstanding programs to the public and to school groups. We have a stewardship and restoration program and that we do a lot of work on sustainable shorelines and looking at projects and how to make sure our tidal wetlands are resilient going into the future. We also have a coastal training program, and that really works to take our scientific information and bring that to the DEC land managers so they can utilize the scientific information in their decision making. So back to our monitoring program, we refer to this as the system-wide monitoring program because all 29 research reserves do the same protocol, so the data can be compared nationally across the system. We do abiotic monitoring for water quality, nutrients. We also have weather towers to get meteorological data so that we could really interpret what is influencing the water quality near our sites. We also do biological monitoring as part of DEC. We're nested in the estuary program's habitat unit. So we look at submerged aquatic vegetation and emergent tidal marsh habitats, both focusing on the spatial distribution and vegetation characteristics. Our monitoring program has been very long-term. We started at all reserve sites in 1991, collecting monthly nutrient data. Starting in 1995, we've been deploying water quality sondes for continuous 15-minute monitoring at Tivoli Bays. When we moved in our facility at Norrie Point in 2007, we started water quality and weather station monitoring there as well. In 2020, one of our pandemic projects was to rethink some of our stations, and we built a new station at Ferry Landing at our northern Stockport Flats component site, as well as started a station at Iona Island at Bear Mountain. And all of these data are publicly available. They're transmitted through satellites in real time. And there's two ways you can access that. Once through the national system, through the Near Centralized Data Management Office. We've been transmitting to there since 2005, and also partnered locally since 2008 with the Hudson River Environmental Conditions Observing System, where you could access some other stations that the Research Reserve doesn't monitor ourselves, but there are many stations along the whole Hudson River estuary collecting water quality and weather data for public and student research use. Like I said, we're nested within habitat protection in DEC, looking at submerged aquatic vegetation. What's pictured there is our native Balissaneria americana, and also looking at tidal wetlands. And one of the ways that we assess the habitat, how it's changing over time, is through a series of habitat maps in GIS. And again, all of those are available publicly through the GIS, New York State GIS clearinghouse with the address included there. So we highly encourage all of the Polgar students, other students doing work on the Hudson and in the Research Reserve to make use of this baseline data as a platform for developing their projects. And if anyone wants any more information about our data sets and services that are available, there's my contact information. So thank you all very much for that. I'm going to stop sharing my screen. And we're going to move right into our student presentations. So up first is Nicole Steinbach. She is from SUNY College of Environmental Science and Forestry. The title of her talk is The Outreach of the Latinx Community in Order to Promote the Preservation of the Hudson River Estuary. Her supervisors are Dr. Valerie Luzadas and Rebecca Hauser. So with that, over to you, Nicole. I can't hear you, Nicole. I'm not sure if anyone else can.
Yeah, no, I also can't hear you.
Not a problem.
There you go.
Very good. Thank you.
Perfect. So what I said before was thank you for the introduction. My name is Nicole Steinbach. I am a junior at the SUNY College of Environmental Science and Forestry. My project is the outreach to the Latinx community to promote the preservation of the Hudson River estuary. And I just wanted to thank everyone for being here today. I'm sure I can speak for my peers and the committee about how excited we are to be presenting to you all today. We've worked very hard this summer, and I'm proud to present the research I've been working on all summer. So slide 2, please. To reiterate the main goal of the project, I want to learn how we can reach the Latinx population in order to promote environmental recreation and education. So to successfully learn to communicate with the Latinx population of the Mid-Hudson Valley, I'm going to talk about my own experience. I am a first-generation Peruvian American. I grew up in the Hudson Valley, and this is really why this project is very important for me. So for those of you working on the Hudson River, it's clear that connecting the community to the environment is an important step in promoting its successful preservation. Thus, creating an environment that fosters stewardship in the individuals of our community is very important and is really why I'm here. So our first step in making it clear, a clear connection is growing up. I mean, for me, a little about myself. Growing up, I had the privilege to attend the New Paltz School District. And it was my time there at the school where I learned about my love for the environment. And of course, this is why I'm here. My school district prioritized taking the students to different preserves and areas for educational programs such as Mohonk, Minnewaska, and the Clearwater Sloop. These places and trips to me were the most impactful ones of my education. And truly, those are the ones that I remember most of. In these field trips, I fostered my love for the environment and my curiosity of the local ecology and my passion to become a steward for the land. This you can see in my career choice as a junior at the SUNY College of Environmental Science and Forestry, and my dedication to becoming a law student and help others experience the environment for generations to come. So this project to me is very important, and I would like to create the educational opportunities that I've been blessed with to experience through my education and spread them to other students like me who may not have the opportunity to go to preserves if not for their schooling. Slide 3, please. So as you can see here, I've listed the main goals of this summer's fellowship. Thus, the purpose of the study is to measure the environmental attitudes of the Latinx population to see if it reflects their environmental recreation pattern, to understand any potential barriers this group has when it comes to environmental recreation and education. As we can see by the hypothesis, a main priority of the questionnaire is to understand the correlation between the group's environmental attitudes and their use of the local environment. This, I believe, is an important step in understanding how to promote stewardship of the Hudson with this community. To do this, the objective of the study are as follows. Of course, the first step in this process was to create a questionnaire that would collect the appropriate data to enable the responses to actually answer the purpose of the study. Thus, we created questions that, when analyzed, will be able to define and assess participation and any perceived barriers by the individuals of this group. And of course, once we hit our target of responses, we will explain and analyze the data to the New York State DEC to shed light on any pertinent issues as a means to improve diversity outreach of this community and generate solutions. Slide 4, please. So in this slide, I will be discussing the project goals and accomplishments. And as you can see, I've listed the goals, accomplishments, and barriers of this summer's project. The goals of this project are fairly straightforward. The first being the creation of the questionnaire, and the second being receiving the green light by the Institutional Review Board to distribute it and use the data collected. So for those of you that are unfamiliar with the IRB, this is also the Institutional Review Board. This is an organization that works under the FDA's regulation to review and monitor any research involving human subjects. So essentially, they work to make sure that your research is ethical and does not breach the legal rights and safety of a participant. So the third goal of the project was to hold a New York State DEC sponsored fishing event at the Norrie Point Environmental Center to bring in the target audience to learn about the local ecology, and of course, since they'll be there, sneak in a little bit of questionnaire distribution. And then our final goal is to collect and analyze our data. So as you can see, our accomplishments reflect some of our goals discussed in the first column. And with any project, are barriers, there are barriers, and one must overcome these. So with this column, I would like to note that the barriers are not final and have served as a great learning curve for me in this process. I'm sure my advisors can attest. These are very important for my study and really reflect the point of what I'm trying to get across. So I'm sure most of my peers, if not all of them, have learned that through conducting the study during COVID-19, there are a few unforeseen challenges, and in my case, this manifested itself in the form of delayed response times for many emails, calls, and meeting up with people. Irb approval as well was also lengthened due to COVID, and among other small issues, many businesses were shut down and closed, so that was also a loss of contact ability and of the ability to distribute and post up flyers. But of course, these challenges did not stop me, and with the support of my advisors, we were able to push through and get ourselves to this point. So another barrier posed is one that I did not anticipate, and is actually the purpose of the study, and that was actually obtaining contact with the target audience. With my background and experience in the area, I believed it would have been a little easier for me to maintain contact with the population, but it actually proved a little more difficult. But of course, this is okay and only serves as a learning curve to push me to delve deeper into how other organizations reach out and remain in contact with the target audience. And at the end of this tunnel, I will come out with learning more, and so will all of you. But more of this will be discussed later in the presentation. So our final challenge was, of course, time. But I would like to think that with any research such as this, time is always more useful and needed. So slide 5, please. So in this slide, I will actually go more into depth about the development and process my team and I went through to obtain Irb approval. For starters, I had to really learn about the proper way to create a science-based questionnaire. Now this might sound silly and might sound really easy for some of you, but you would be surprised about the almost common sense things I've learned while creating one. So coming into this project, I did have prior experience and research done with this study. What I mean by this is I actually spent my previous winter working with the DEC to understand academic literature about the culture, environmental ethics, and attitudes of the American Latinx population. And of course, along the way, I also learned about some theories that help explain why certain barriers occur more frequently than others. So research can give you many answers, but it cannot create the questionnaire for you. Thankfully, I had the opportunity to work with Valerie, who was very well experienced with research creation, implementation, and you might have guessed it, questionnaire design. So she has served as an important aspect in me understanding how to develop my findings of theories and create a questionnaire that not only embodies the work I've put into understanding these phenomena, but when analyzed, these responses will refute and or confirm these theories. So I'm going to skip ahead to the last bullet point to answer the ones before it. But Irb approval initially was not something I was previously familiar with. Thankfully, with the help of Valerie and her experience, and with the conflict of the process being prolonged, and the conflict of the process of Irb approval being as prolonged as it was due to COVID, this was not as challenging as it probably should have been. So for those of you unfamiliar with it, the process involves an application packet of sorts. This application proved to be, at least in my opinion, more useful to me than for the actual review board. This process enabled me to physically map out exactly what I wanted to achieve with the questionnaire, how I was going to implement it, and to really delve into what I would be asking from the participants. And as funny as it is, it actually helped me really plan on how I was going to recruit people. So this experience truly enabled me to stay organized by outlining my project and with fine detail, and of course to stick with my initial goals and what my actions were. So slide 6, please. In this slide, I will discuss the layout of the questionnaire. As you can see, there are 7 sections assigned with different purposes. As I mentioned in a previous slide, I had researched several theories to understand the pattern of Latinx environmental attitudes and their participation in environmental education and recreation. Interestingly enough, there are theories to understand why Latinx people are typically not as involved in environmental recreation and education. Thus, Each of these listed sections were created to group questions together that reflect certain theories. So I'll briefly run through these sections. So section 1, as per IRB instruction, this section is concerned about the rights of participants and is an explanation of what the data is going to be used for. And so essentially there's a blurb about the questionnaire, who I am, and why the data is going to be collected, and a waiver of consent. In section 2, we delve into understanding the participants' attitudes about the local environment and their familiarity with the generalized areas programs, and educational opportunities that are available at the surrounding state parks. Section 3 goes into understanding their utilization of the state parks, whereas section 4 delves into if their utilization reflects their desire to participate in their local environmental recreation and education opportunities. This is used to shed light on any perceived barriers they may experience when doing so. Now, section 5 is actually a pretty neat one, and this one solely aims to understand their environmental attitude. This is done by using the new ecological paradigm scale. And for those of you unfamiliar with what this is, it is essentially a list of about 10 statements that participants are asked to essentially agree or disagree. They're asked at what level they agree or disagree with each statement. Each response is attached to a number that, when all the responses are combined, will illuminate one's pro-environmental and/or anti-environmental attitude based off of a final score. So now section 6 serves as our understanding of the participant. Here we want to know about their age, their cultural influence, among other details that may produce common patterns in the collected data. And in section 7, this serves as a way the participant can contribute their ideas and thoughts about the participation in the questionnaire. So maybe we missed something that they may perceive as an important barrier, or maybe they didn't like a question. So here we essentially get the opportunity to learn from them. So slide 7, please. So here is just a sneak peek at a couple questions in the questionnaire. As you can see, these are actually 3 questions drawn directly from the document. Later on, I will send a link for those of you interested in looking over the entire questionnaire. But to go through these very briefly, a question from section 2 listed here, talks about how connected to the local environment do you feel? And the responses can be a great deal, a lot, moderate amount, a little, or none at all. And in section 5, there is the new ecological paradigm scale. And as you see, the statement on the left is plants and animals have as much a right as people to live. And the participant can pick their option of response, and it could be strongly disagree, agree, or strongly agree. Slide 8, please. So here is the implementation plan. Here we can see the plan and the steps I actually took to distribute the survey and spread the word about the project I'm working on. The implementation part was actually a very fun and interactive experience for me. And I'm not sure if this has come across yet, but I've had the privilege of having a very strong support system with my advisor, Valerie, my friends over at Norrie Point and the DEC, as well as my friends and family and other people that I've met along the way. With that said, to actually distribute my flyers, I drove around with 2 of my friends and went from place to place, city to city, putting up flyers at shops, restaurants, markets, churches, grocery stores. If it was a hub for the Latinx population, I was there. In doing that, to my friend's surprise, I also went up to people and handed out flyers. I think this was one of the most impactful experiences because the things you can learn from random strangers can really give you perspective. What I've found is that people hold different stories and truths that won't come across on paper. And what I can say is that the population is very eager to help their youth, and they want to have more programs for their children. You know, I thought I would encounter some challenges and many no's while distributing and posting my flyers. But as soon as I mentioned anything about the youth and helping them learn more about the local ecology and create more programs tailored to them, it was a done deal, and I had clearance to post my flyers. A popular phrase used was anything for the children, and they really meant that with conviction and excitement. So in the next 2 slides, I'm going to actually show you about a few of the implementation steps. These will be the flyer and the event that I held as well. And I'm going to talk more into depth about these. So slide 9, please. In this slide, we can see the original flyer I posted regarding the survey. A brief translation of this at the top in white, we have questionnaire, and below that we have participate in a questionnaire that can help everyone in the community. Then below this, it says to scan the code, which will then direct the participant to the survey. Below that is simply a brief summary of who I am and what the research is about. As I previously mentioned, these flyers were distributed in various locations in New Paltz, Kingston, Poughkeepsie, Walk Hill, and Hyde Park. I handed out flyers to people along the way as I saw them. I posted them in restaurants, in local grocery stores, local convenience stores, churches, youth centers, among other places as well. So slide 10, please. So now we're going to talk about the fishing event. This was a New York State DEC sponsored and marketed fishing program held at the Norrie Point Environmental Center in Stratford on August 2nd. This event was created to draw in Latinx families so that children could experience and learn about the local ecosystem, while I also got to distribute the questionnaire along the adults of the families. So it was a win-win in my opinion. But this event was very marketed, and when I spoke to people about it, I was faced with a lot of excitement and enthusiasm. So of course, I took this well and expected a successful turnout, but to my shock, this was not the case. We encountered one family, and as ironic as it was, they had actually not known about the event, which was heartbreaking to me because I went everywhere to get these events out and distributed it as much as I possibly could. I was really excited. But nonetheless, this family was very excited to participate in our program. So when talking to one of the parents, we mentioned our project and some other environmental education programs that are offered to the youth in the area. The most pertinent one we talked about was the Tides program held at the Norrie Point Environmental Center, which I also had the privilege of helping out at. So the parents' interest in recreation, environmental recreation, to our event and the other events was very heartwarming and really the reason why I wanted to conduct the study in the first place. The father discussed his interest in taking his children to local environmental education programs and how he believed it would serve as an important aspect in his children's education. Yet he did not know where to access information about these programs. So this interaction really depicted how meaningful this project is, not only to me, but to the community. And this experience to me was not a deterrent, rather it was an opportunity to learn and grow. It embodied my main question, why environmental attitudes, and in this case, the desire of environmental recreation, does not reflect the population's participation in such activities. Thus, more work must be done to reach this audience and create opportunities for them to learn about and participate in environmental education programs. So this leads me to the next slide, actually. So slide 11, please. So what are our next steps? How can I use my experience from the summer and create successful outreach solutions? Well, I'm going to determine new ways to reach the target audience. This can be in the form of talking and learning from different people and or businesses that do this successfully, such as churches or community centers or even magazines. And then, of course, I will also be implementing the newfound approaches in order to create a more successful outreach program for the New York State DEC that targets the Latinx community. So slide 12, please. So to wrap things up, I just wanted to say thank you to the Polgar Committee for funding and believing in my project. Thank you to Fran Dunwell for helping me learn about the Hudson River Estuary and the many opportunities in this community creates for young people like me to experience what it's like to work in this field. And of course, a special thanks to my advisors, Valerie and Rebecca, who have been great resources for me along this project's journey. So now I will ask, are there any questions?
Thanks, Nicole, and I will help her facilitate the question period. We still have about 10 minutes. If you want to type your question into either the chat or Q&A or use the raise hand function, I will call on you. I don't see any hands raised currently.
Hi, Nicole. I have a question, actually. I don't know if you're familiar, in 2018 or 2019, a bunch of researchers in Detroit put together a tree planting program for the minority groups in Detroit, but there was a lot of pushback and resistance, and the researchers kind of found out that it was due to the minority groups not being involved in the decision-making process. So these researchers planned this event, but then there wasn't any turnout because they weren't involved in planning the event. So do you think in the future you would have the Latina community be involved in planning the event?
So that's actually really interesting, and a lot of what my actual classes I take at school go through with minority communities and them getting taken advantage of very easily because there aren't support group systems or centralized areas for them to voice their opinion. And I personally would love to see and help facilitate this once I understand and finish my project, of course, how to really reach this community and how to keep them together and make them more, help them participate more in community stewardship, so in other educational programs, and really in like the policies. There are a lot of community efforts to talk about the policies. A lot of people want to hear what the community thinks about certain policies that get planned, and I would love to see them being able to participate in that. And I have some ideas of how to do that, and with learning how to reach this target audience, I would love to help implement them as well.
Thanks, Nicole. I don't see any other questions in the chat right now. If any other panelists want to jump in with a question, go ahead.
I just want to encourage the audience, in case they're feeling a little bit shy, that we are encouraging questions from the audience. So you don't have to feel shy if you have something, and feel free to write it during the presentation, because we'll pick it up in the chat afterwards.
I'll ask a question. Nicole, do you feel that the IRB process took up like a disproportionate amount of your time and affected the trajectory of your project? Or did you kind of account for that? Because you said how it was a long process, you weren't sure about it, you had to like kind of learn as you go. And I know from experience, it is a long process. Did you feel like that kind of threw you off, or did you kind of plan ahead for it being a long time?
So it was a lot longer than wanted to get approval, because of course I could make the questionnaire, I could be ready to distribute it, but until I get the final say, I can't really do anything. So we did have it set up where I was going to go, what I was going to do to actually distribute it, and we had the plan about the fishing event. But of course, going places and giving this out, distributing it online, and really getting it out to people had to wait until approval. And I think it did take about roughly a month. So it definitely did take a disproportionate amount of time in comparison to actually implementing my project, which was very unfortunate. But I think we are trying to do a really good job of doing as much as we can now and getting it out as best as we can, regardless of how long it took to get approval.
Thanks, Nicole. Any other questions? Anyone seeing anything in the chat that I'm not seeing? Oh, maybe one came up here in the Q&A from Margie Turin. Was the questionnaire provided in Spanish or in English?
So I made 2 copies of the questionnaire. I did a English version for my peers and colleagues to be able to read easily in the event that they did not speak Spanish, but the questionnaire and all of the flyers themselves were actually provided for in Spanish. But of course, not all Latinx people can fluently read Spanish, so that the English questionnaire as well can be distributed to those who would need it in English. So there were 2 copies, but the Spanish one was predominantly used. I hope that answered the question.
Sounds good to me. Another one just came in from Laurel Zima. Hi, Nicole, fantastic project and presentation. I was wondering if you performed the questionnaire with the participants at the fish count before they got their hands wet and handling the fish, etc. I just wondered if that interactive fun and hands-on experience could have impacted their results.
So the children themselves did not take the survey. For IRB approval, we made it so that only 18 and older adults could actually take the survey. That was just a much easier way of getting approval faster and not having to go through the hassles of being able to get children to take it. And the parents also probably have a little more insight and more ability to really like take the survey and understand what we're trying to ask for. So essentially, the family that did come in had a couple small children, and they came in to actually see the fish tanks. So they got a nice little personalized tour of the fish tanks with Rebecca's help. They also got to feed the fish, and which was really neat. And they got to go seining, which was very fun. And I think that was their first experience with seining, and their parents were very happy with it. And the parents, they took the questionnaire later on, I think, in that day. I gave them a flyer with the scan code so that they could take it on their free time and they could enjoy their afternoon with their children. So I don't think it would have impacted their results, if not illuminated them to the opportunities of children actually being able to experience hands-on learning and how fun the local environment actually is. But I hope that answered the question.
Thanks, and I just want to note that Valerie typed in that their usual IRB process at ESF is about half of the time than it took for Nicole. So it did sound like there was some COVID impacts going on there. Anyone else? No open questions at this time. I'm not seeing anything in the chat. Okay. Well, thank you very much, Nicole. Let's move on. All right. Let's move on to our next presenter, which is Royal McMahon Ward from Fordham University. She will be presenting on microplastics exchange between the American horseshoe crab and the Western Hemisphere shorebirds through horseshoe crab eggs in Jamaica Bay. And her supervisor is Mark Botten. Royal, whenever you're ready.
Awesome, thank you so much. So hello, my name is Royal McMahon Ward, and today I will be presenting on my research project that I conducted this summer under the advising of Dr. Mark Botten that is titled Microplastic Exchange Between the American Horseshoe Crab and the Western Hemisphere Shorebirds Through Horseshoe Crab Eggs in Jamaica Bay. So the field site for my project was Plum Beach, located in Brooklyn, New York. All of my data was collected from the eastern end of Plum Beach, where beach conditions are optimal for horseshoe crab spawning. This results in a higher concentration of horseshoe crabs and thus horseshoe crab eggs in this area. The American horseshoe crab, or Limulus polyphemus, is a marine arthropod that inhabits the Atlantic coastline of North America, extending between Mexico to Maine. Limulus polyphemus spawns on estuarine beaches, such as Plum Beach, where it lays its eggs. Horseshoe crabs are a key species in the estuarine food web, where its eggs provide an essential food source for a number of migratory shorebirds. Horseshoe crab eggs are fertilized externally, where a male horseshoe crab will form amplexus with the female horseshoe crab during the spawning season in May and June. The female horseshoe crab holds its eggs inside its prosoma, as shown here. And in fact, when the prosoma of a female is punctured, which is common during tagging, and it is during spawning season, the female will have eggs come out of the puncture wound. Horseshoe crab eggs are laid in the sediment of estuarine beaches by the female horseshoe crab, where most batches of horseshoe crab eggs are laid approximately 8 to 10 centimeters deep. These eggs will stay embedded in the sediment until they develop and hatch out into their larval stage later in the summer. So Jamaica Bay, and specifically Plum Beach, is a staging area or an intermediate stopping point along the Western Hemisphere shorebird's flight. The high abundance of horseshoe crab eggs in this area serves as an important feeding site for shorebirds along their migration. Horseshoe crab eggs are the dominant and near exclusive food type for the semi-palmated pulver and the semi-palmated sandpiper, as shown on this slide, which are two of the most abundant shorebird species in Jamaica Bay during late May and early June. This was discovered by a master's student at Fordham in 2019 named Emily Casper, who found this based on a DNA analysis of the shorebird fecal pellet samples collected in Jamaica Bay during those months. So a little overview of this ecological link is that we see that horseshoe crabs feed by consuming organisms that are embedded in the sediment. These horseshoe crabs then lay their eggs in the sediment on these estuarine beaches, and migratory shorebirds, such as the semi-palmated pulver and the semi-palmated sandpiper, consume those eggs along their migration. This important ecological link is threatened by the increasing presence of microplastics in Plum Beach, New York, and in the marine ecosystems in general. Microplastics are an ever-growing concern for our planet, including our estuaries and oceans, where it has become the most abundant type of anthropogenic debris. Microplastics enter the marine ecosystems in many ways, including litter on beaches, runoff, and the direct release of sewer water containing high amounts of microplastics through combined sewer system, all of which impact the New York area and the Hudson River estuary. The impact of microplastics can be great, as plastics can transfer up the trophic chain and have been known to absorb chemical pollutants, which raise concern over how these chemicals may be moving through the food chain and impacting these marine species. In Jamaica Bay specifically, it has been noted that the WWTP outfalls and CSOs are the primary point sources for microplastic contamination for Jamaica Bay. Some sources of primary microplastics that are prevalent here originate from cosmetics and laundry services, and some secondary microplastics come from the weathering of larger plastic debris, which can be found as plastic trash on the beaches or just in the sewer system in general. So for my research project, I looked to assess the possible transfer of microplastic pollutants from horseshoe crab eggs to shorebirds, and in addition, determine if microplastics are present in the eggs of Limulus polyphemus, either through the maternal transfer of microplastics during oogenesis or environmental exposure once the eggs are laid in the sand. So during this summer, we collected all of our data from the field on June 7th. We collected horseshoe crab eggs in two separate groups. The first group of horseshoe crab eggs that were collected were from the female horseshoe crab before they're ever exposed to the surrounding environment. We walked on the tidal flats of Plum Beach East and selected 10 females, collecting 20 to 60 eggs through the stimulation of the genital pores shown here. Due to the unusually early spawning season that we witnessed this year, some of the female horseshoe crabs that we were able to find on the beach did not have enough eggs to generate a large enough sample solely through massaging their genital pores. But due to the branching ovaries that female horseshoe crabs have, which allow for their eggs to be free floating in their prosoma, we were able to collect a few additional eggs during the tagging process that were taking place on the same day. So before the tag was placed into the puncture wound on their prosoma, we were able to collect additional eggs to complete our sample. All of our eggs were then placed in a small glass vial shown here in this image. The second group of horseshoe crab eggs that were collected were eggs that were laid by the female horseshoe crab and were exposed to the surrounding environment for up to around 3 days, approximately. We collected eggs from the intertidal zone on Plum Beach East, where we collected 40 to 100 eggs from 10 separate batches. And these eggs were collected with a metal shovel and placed into glass jars. We also collected water and 2 separate sand samples on the same date. We collected 1 liter of beach water during low tide in a glass bottle, and we collected sediment samples from the tidal flats where we found our female horseshoe crabs, and we took sand from the intertidal zone where we collected our laid egg samples. Shorebird fecal pellet samples were previously collected in 2019 by Emily Casper, a Fordham master's student, and were stored in glass evaporating dishes covered with tinfoil to prevent contamination. So moving to the lab work, we utilized a Nile red staining protocol. Nile red is a red fluorescent stain that binds to neutral lipids, which makes it suitable for discriminating microplastics from sand particles, which was very important for our research. But because Nile red binds to neutral lipids, this means that it would bind to organic material as well as microplastics. So in order to accurately detect microplastics using Nile red, all organic material first had to be removed. Before we treated any of our samples, we wanted to create a positive and negative control to ensure that our Nile red staining protocol was working. So for our positive control, we created plastic particles using a large metal file on a plastic water bottle lid. These particles were then stained with Nile red and viewed under the microscope. In the lower right-hand corner, we can see those plastic shavings under the bright field, and then next to it, those plastic shavings under the green fluorescence. We also created a negative control using Plum Beach sediment samples that was heated to 500 degrees centigrade in a muffle furnace for 5 hours, where it was then allowed to cool before being transferred into a cleaned glass jar and stained and viewed under a microscope. So in the lower left-hand corner, we can see the muffle furnace sand under bright field and then the muffle furnace sand under green fluorescence. And it's notable that we note no fluorescence when exposed to the green fluorescent wavelength. These tests ensured that our Nile red staining was working as it should be. So in order to generate a manageable sample to stain and view for our sediment samples, we took 20 grams of each sediment sample and added 80 milliliters of 73% NaCl solution. This was placed on a stirring plate for 10 minutes. The particles that were suspended in the solution were carefully poured over a 125 micron sieve, where then an additional 80 milliliters of the NaCl solution was added back to the beaker, placed on the stirring plate for another 5 minutes, and then the rest of the solution was poured over the same sieve to generate our sample. This allowed us to separate the bulk of the microplastics from the excess sediment so that we did not have to process all of that sand and then stain it. We also poured the water sample over the same sieve to separate out any microplastic debris. Each sample was then transferred to a vial and placed in the hydrogen peroxide solution. So to remove all of the organic materials and to make sure that all we had left were plastics and sediment samples was that we treated them all with 30% hydrogen peroxide in order to oxidize the organic material. For the horseshoe crab egg samples, we used 20 eggs, approximately 85 milligrams per each sample. This was deemed a suitable number to be able to generate some useful results. These samples were then placed in 60 degrees centigrade water bath for 24 hours. The samples were then transferred to a clean watch glass and placed in a 60-degree oven for 36 to 72 hours until the hydrogen peroxide was fully evaporated. At that point, once the samples were fully dry, they were then treated with approximately 4 drops of Nile red and given between 30 minutes to an hour wait, which was deemed the appropriate time for the stain to work before being viewed under the microscope. Then each sample was viewed under green fluorescent at a wavelength of 488 nanometers at 40x magnification. We randomly selected a 2 by 2 millimeter area where in 4 horizontal transects, we took 5 photos per transect, totaling 20 photos per sample. Each of these photos was then processed using image J to count the total number of particles and to size the average particle per each photo. For image J to be able to size our particles accurately, we had to measure our microscope area using a stage micrometer, which measured to be 2.2 millimeters wide. So on our slide here, we see that the top photo is a fecal pellet sample under green fluorescence. And below that, we see the same photo after it has been processed with image J. We were able to generate some basic results thus far. First, we were able to generate an average number of microplastic particles per sample for our fecal pellet, unfertilized, fertilized egg, tidal flat, beach sand, and beach water. And we see on the y-axis are different sample labels, and on the x-axis, the average microplastic count. Our graphs are separated because we had a different number of samples per each of these. We were only able to collect and process 1 sample per each sediment and water, whereas we had 10 samples of each fecal pellet egg sample. We were also able to generate some results when it came to the average microplastic particle size per each sample, where on the x-axis on these graphs, we have the average size in millimeters squared. Again, they are separated based on the number of samples that we have been able to process up until this point. Some tentative conclusions that we have found is that first, microplastics are present in all of our samples. No matter which sample we looked at, we did find microplastics. We also found that since microplastics were found in unfertilized eggs, this indicates that microplastics do in fact transfer from the female horseshoe crab directly to the eggs during oogenesis. We also noticed that the unfertilized eggs had higher average microplastic count compared to fertilized egg and shorebird fecal pellet samples. We also found that the average microplastic size ranged between 0.224 micrometers to 1.22 micrometers for all of our samples, and that the average microplastic particle size was largest for the fertilized egg samples. So some further work needs to be done to finish up this project. The first includes completing statistical analysis of the differences in the number of microplastic particles and average microplastic particle size, comparing unfertilized eggs, fertilized eggs, and fecal pellet samples using analysis of variance. And finally, we have to complete additional replications of the sand and water samples to provide more data to be able to generate better conclusions. Finally, based on the preliminary results, microplastics appear to be more mobile than previously thought. Due to the increasing prevalence of microplastics in the marine environment and the possibility of bioaccumulation and biomagnification of toxins within marine life, further study into how microplastics in the marine environment may impact horseshoe crabs and their development is needed. Finally, I would like to thank Fordham University for their use of their lab and equipment, as well as the Hudson River Foundation for their funding and support of this research. And in addition, I would like to thank Dr. Rodanis Ruano for her instruction on the fluorescence microscope. And lastly, I would like to thank Dr. Mark Botten for his support in this project in summer. I could not have done this without his help. Thank you.
Thank you so much, Royal. We already have a couple of questions for you on the chat. The first one came in from Marty Rowland. He asks, is there concern for sacrificing horseshoe crab eggs? Are they a threatened species?
So there's no concern over threatening the population of horseshoe crabs by collecting eggs. We did not collect that many. If you look at the total number that are laid by the female horseshoe crabs at Jamaica Bay, it is less than 1%, definitely, maybe even less than that. As well as horseshoe crabs produce eggs, produce enough eggs where most of them do not survive out in the wild anyways. And so our sample size would not have an impact on their population.
Thank you. Judith Weiss asks, what shape of microplastics was the most abundant in your samples?
Jagged oval shapes was the most prevalent, I would say. None of them were perfect circles or a perfect geometric shape. Most of them had pretty ragged edges, but most were circular.
I don't see anything else in the chat right now. Do any panelists have any questions? We've got a couple more in on the chat. Luana asks, how were you able to completely separate the organic matter from the inorganic matter before the staining protocol?
So that was where the hydrogen peroxide solution comes into play. 30% hydrogen peroxide solution for 24 hours at 60 degrees centigrade sped up the oxidizing process, which removed all organic material and leaving microplastic and sediment unimpacted. And that is the process that we chose to use. It did require some trial and error to get the timing correct so that we could be sure that there was no organic material present.
Thank you. And Carla Gonzalez asks, or says, hi, Royal. I was wondering what steps can be taken to reduce the amount of microplastics found in horseshoe crab eggs?
I don't know specifically in terms of how we would prevent them from just horseshoe crab eggs. I think an important step would be to reduce the amount of microplastics that are put into the marine environment as a whole. Due to the high abundance that is found in the Hudson River estuary, as well as most marine ecosystems, it is going to be hard to limit the amount that horseshoe crabs are exposed and thus able to transfer to their eggs. One thing that New York City could do would be to change their combined sewer systems so that there isn't excessive runoff during storm surges. That would be, because those are marked as point sources for Jamaica Bay, that would be a great starting point to reduce the impact.
Ray Kepner asks, any ideas on mechanisms of microplastic transport into ovaries or transport into cells of primary oocytes?
As of right now, I do not. I have been trying to generate a better understanding of the development of horseshoe crab eggs within the ovaries of horseshoe crabs, but there is not extensive literature in that area. So as of right now, I don't have a great understanding of the exact mechanisms that it would take place. But I hope to have a better understanding moving forward.
Laurel Zama says, great job, Royal. Your results are fascinating. And the shorebird fecal pellets, is there any way to differentiate whether the shorebird consumed the microplastics from the eggs or by mistaking microplastics for eggs directly, like in the size?
There's no way for me to know whether these shorebirds are able to distinguish them. But if we look at the general behavior, these shorebirds are feeding in this area in a specific way in order to consume horseshoe crab eggs. So whether or not they're eating the eggs and getting microplastics through the eggs themselves, or through the sediment that is attached to the eggs, or by mistaking other microplastic particles for eggs, the behavior is the same. So we do know that microplastics were present in the eggs themselves, but they could be consuming additional microplastics through trying to consume these eggs as a whole.
Great. There's also a bit of a side conversation going on in the chat about microfibers. Judith Weiss responded that the most abundant microplastics in the environment are microfibers, so your method must have missed them. They are long and thin and may not have picked up the stain. And then Jane Alexander asked if the microfibers might also be lost through the filter.
So we did not see any we did not see as a main microplastic contributor for the egg and fecal pellet samples to be fibers, but in our water and some of our sediment samples, microfibers were more prevalent. So I think it just kind of depends on what you're looking at. It seems that microfibers may not be be found inside of the horseshoe crab eggs, even though they are a prevalent microplastic in the marine environment as a whole. But hopefully when we collect more water and sediment samples, we'll be able to have a better understanding of how microfibers make up the total microplastic contribution.
Thank you so much. I don't see any other questions in the chat right now. Any other questions? All right, I think we'll stop it there, Royal. Thank you so much for your presentation and answers.
So welcome back, everyone, for the next session. And the next session will be moderated by Dr. Yazzo, and I'll turn it over to Dr. Yazzo. Thank you.
Thanks, Helena. So our next presenter is Emma Butzler from Marist College, and her presentation is entitled Understanding the Geologic and Economic Controls on Drainfield Effectiveness of Septic Systems in Hudson River Tributaries, and her supervisor is Zion Kloss.
Okay, can you guys all hear me?
Yes.
Okay, so hello, my name is Emma Butzler and my advisor is Dr. Zion Kloss. And I'm excited to share my research titled Understanding the Geologic and Economic Controls on Drainfield Effectiveness of Septic Systems in Hudson River Tributaries. And this is a socio-environmental approach to addressing excess nitrogen and nutrient loading on the Hudson River. So a little bit of overview of what I'll be talking about today. I'm going to be talking about nutrient loading and how it relates to harmful algal blooms on the Hudson River, the location of my study and the unique soil and geologic properties that are seen within my location. I'm going to give a little bit of background on decentralized septic systems, specifically focusing on drain fields. And then that will lead me into my research question. And then I'll talk about my experience this summer, what I did, how I analyzed my results, the geospatial maps that I created, what I'll be doing into the fall. And then I have a little bit of a reflection of my past summer. So nutrient loading on the Hudson River is a growing concern because of harmful algal blooms. They threaten the ecology, the economy, and people. Excess nutrients in the form of nitrogen and phosphorus are the main contributors to these blooms. So there's a necessary need to investigate tributaries as possible points of this excess nitrogen. So the tributary that my study looks at is the Falcao Watershed located in Poughkeepsie, New York. And this is considered a Class C stream. The headwaters start in rural areas, traverse downstream into rural Hyde Park, and then flow into the city of Poughkeepsie before discharging into the Hudson River. And what's unique about this location and this watershed are the soils, the geology, and the amount of decentralized septic systems that are seen within this watershed. So according to the USDA Dutchess soil analysis, the majority of the soils are excessively drained, the water tables are high, and there tends to be faulty bedrock. And this means that nutrients and other bacteria can quickly make it into the nearest water source. So how does this apply to decentralized septic systems? So a decentralized septic systems is an on-site wastewater treatment system where the wastewater on the property is treated and disposed. And there's two parts, the septic tank and the soil drain field. And I'm specifically focusing on the soil drain field for my project because it relies heavily on the soil. And how the drain field works is the wastewater enters the drain field and percolates downward into the soil. And the specific chemical and compositional characteristics of the soil acts as a natural filtration service, as well as the bacteria. It breaks down any kind of contaminants that are in the soil. And then if working properly, the soil should filter the water properly, that the water that's released into the water body should be treated. So how does this apply to my site specifically? As you can see, this map on the left, the watershed is scattered with a lot of decentralized septic systems. And you can see that the yellow is excessively drained soils, and a lot of the decentralized septic systems are in location of excessively drained soils. The drain field requires moderately to well-drained soils to be considered effective, and there needs to be there can't be shallow soils either. There needs to be a certain depth, and there needs to be a specific percolation rate. So excessively drained soils aren't considered suitable for the final treatment of effluent in the drain field. So decentralized septic systems, it's the homeowner's responsibility to maintain or replace the sewer because it's on their property. Therefore, it's their job to pay for any kind of maintenance or replacement cost. And as you can imagine, something like that can be very costly, especially replacing septic systems. So both of those factors, understanding the soils and the geology, the faulty bedrock, and the amount of decentralized septic systems in my location led me to my research question, which is, are the geologic and economic controls on septic sewers in the Falcao Watershed contributing to excess nutrients that are ultimately reaching and affecting the Hudson River in forms of sinohabs? So using GIS and in situ measurements of nitrate, the goal was to spatially understand locations of possible excess nitrogen from these faulty drain fields. A large part of my research was working with GIS. So the first thing I had to do was understand what was happening on the landscape and under the soils. So I first had to make some maps understanding the soil. And I used the USDA NRCS soil data for Dutchess County, and this is public data. And I removed any soils outside of the Falcao watershed, and then I grouped the drainage class of soils to illustrate the unsuitable soils. One thing to mention is that the researchers that were doing the data analysis had already classified what was considered excessively drained, poorly drained, moderately drained, or well drained. I personally did not decide what was considered what. I just grouped the classes together to create this map. The second map that I created using the Dutchess County tax parcel data, which is also public information, I removed any properties that were on a centralized system, so I could only look at properties that were considered decentralized septic systems. And then to show the economic data, I built an expression because income level is not something that is publicly accessible. I built an expression where I took the property value and divided by the square footage of land to give me a rough estimate to reflect the ability to maintain and replace septic systems. So the second part of my research was actually going out in the field and collecting nitrate data and locating those possible sources of nitrate spikes. So using Google Maps, me and my advisor, Dr. Zienkloss, decided we wanted a high resolution data set. And to do that, we wanted to take nitrate values every 50 meters. So I mapped out the entire Falcao, well, minus some tributaries, but the picture on the left is a bit ambitious. There's 600 data points. And I did try to get some tributaries, but not all of the Falcao was walkable. So I ended up getting 400 data points. And to put that into perspective, 400 data points every 50 meters is about 12 miles. So I got 12 miles worth of nitrate data. And me and Dr. Zion decided that there needs to be a minimum of 0.33 inches of rain because previous studies had shown higher variations of nitrate after rainstorms or in wet conditions. Luckily, the month of July was one of the rainiest months on record. So reaching that minimum was never a problem. So I used the electronic YSI probe that measured nitrogen, conductivity, temperature, chlorides, and dissolved oxygen. And I collected every 50 meters. And then every 150 meters, so every 3 data points, I took a water sample that is going to be sent down to Dr. Zarnak's lab in New York City, and that will be tested for other forms of nitrogen as well as phosphorus. So once I get those tests back and that data set back, my data will be a little bit more stronger, and I will also have phosphorus to add to some of my maps. And so the way I analyze my results is every data collection day, I collected within points. So 1 through 39, 40 through 85. And I had to analyze that data set within the data set, just because every day was so different depending on how much it rained. So I took that raw data and I normalized it. And when I normalized it, I took the raw data minus the day average divided by the day of the standard deviation of the day. And what I noticed from all of my data sets was that there was this slight upward trend from my YSI probe. So we decided to normalize it again. Basically, we made a trend line and made that trend line the x-axis to see spikes and troughs and variation in the nitrogen. So the way I normalized it again was I took the distance from the trend line, I took the normalized data and then subtracted it from the distance. And that's how I got the nitrogen variation that I used for my geospatial analysis and what you will be seeing for the maps that I will be showing you. So the way I decided to present my data was using Esri GIS online, and I created an interactive map, and I wanted to make all of this data publicly accessible for researchers, planners, anyone that might be interested in what's going on within the stream. I have a description of every single value. I also have a description of how I got the nitrate variation using the raw data, so everything that I just went through. And then I also have sites of future FIB tests. And so I'm gonna put this link in the chat real quick. Okay. I'm also gonna share my screen. I added a QR code. Okay, can everybody see that? Okay. Sorry, did you say everyone can see that? I just want to make sure, Sarah.
Yes, we can.
Okay, perfect. If it starts being glitchy, just let me know, and I have what I'm going to present on some of my slides. So I want to go through how to use this interactive map real quick. I have all of the layers on this map, and the layers are the fecal indicator bacteria test, the nitrate variation, where the on-site septic systems are, and then the stream, and then the picture of the Falcao Watershed. And I have the legend, and the way I decided to quantify the nitrate variation was using basically a box and whisker plot. So any data that was an outlier wouldn't skew the data. And then I have a little about page so you can understand exactly what you're looking at. You can turn off any layers you want. And then I also have this infograph. And so this infograph is where you can see where exactly the highest spikes are occurring. What this is all of my nitrate variation data on one graph. So right here would be a spike, and it correlates with the 0.139. And you can see that it outlines in blue. So you can go into that section and see, oh, what's happening on the landscape, you can turn that off, and you can see that this is a possible spike in nitrate, especially looking at the infograph, it's very high. And because this is a possible spike in nitrate, this is going to be a future site to test for enterococci forms. You can also change the base map so you can see and understand what's happening on the land. You can change it to imagery to see if something is more residential or if something is more in a forested area. And then I also have each of the data points. I have each of the data points for each of the days I collected that you can turn on and off. So this was the first day I collected points 1 through 39, points 40 through 85. So you can see specific days. And then I also have a share button, and then I have an analysis button. Oh, I don't know what happened. My bad. Hold on. Did that turn off on everyone too?
Yeah, we can see your story map right now.
Okay. Sorry, I've been having difficulties with, give me a second. I've been having difficulties with technology this morning. Sorry about that. Okay. So I'm going to go through some of the sites that I thought needed further tests for enterococci count. And the goal with me going through the Falcao stream to locate possible sources of nitrogen was to go back through and see if it correlates with the decentralized septic system. So right here is a perfect example. Can everyone see my screen? Just want to make sure.
Yeah, looks good.
Okay, perfect. Okay, so you can see right here, there's a spike in nitrogen level, and it's very close to decentralized septic systems. So going back through to test to see if this spike in nitrogen level is from fertilizers or from faulty decentralized septic systems is the reason why I'm doing the FIB test, to see why exactly the nitrogen is spiking. Some other spots that I'm going to go back and test for in the fall are right here. And you can see that in relation to the decentralized septic systems, there's quite a lot, and it spikes really high right here and then goes down and spikes again right here. So these are the spots where I decided to test, especially coming right after a tributary. I think it's interesting that it begins to spike after a tributary, especially seeing where the tributary lands in relation to the decentralized septic systems. Other spots that I'm going to test are right here. This is point 285. And I think I go all the way up to Camp Victoria Lake. Like I'd mentioned, all the data is on here. So if you click on a data point, you'll have the latitude, the longitude, the nitrate variation, and then all the information that I talked about. There's also the Falcao stream information, and then you can also find information on the soils, and it gives descriptions on the specific soils you're looking at. So every soil is a little bit different. Yeah, so if you guys want to go check it out when we're done with this, there's a lot of good information on here. And the idea is for researchers and planners and the Falcao Watershed Coalition to be able to look at this and understand possible points of faulty decentralized septic systems. Of course, the FIB test that I'll be collecting in the fall, I'll add this and make everything publicly accessible. And the nitrogen and phosphorus data that will be tested in Dr. Zarnock's lab will also be added to this data set. So back to my presentation. Okay. Sorry, how do you start from the slide you're on? Slideshow? Oh, there we go. Okay. Like I had mentioned before, future directions, I'm going to be using the water samples from Dr. Zarnak's lab will give me more information on phosphorus and nitrogen. And then the enterococci counts, there's about 18 spots that I'll be testing. Thanks to Dr. Kepner, who said he'd be willing to do those tests for me. And then testing possible tributaries would be something of interest. And then the last thing I want to go through is a little bit of my reflection from this past summer. And I made a story map, and I'll also send that in the chat, but I won't share my screen because it's not going too well. Okay. So I added the link in the chat. So of course, walking 12 miles through a stream, you're going to meet a lot of people and see a lot of things. And it's really cool. It went through the city of Poughkeepsie and then into Royal Hyde Park. And what I've realized is that this stream is a huge part of people's lives in different ways. So this picture on the left, going through the city of Poughkeepsie, kind of looks like that city in Italy. But one of the women I met, I had to recollect this site twice because I had taken one of my business major friends out and I didn't fully explain how to fill up a vial of water. So I had to recollect in that spot to get those vials of water. But the first time I went through this portion of the stream, I met this woman named Stacy, and she was peering out the window and with her cat. And we had a little conversation. I explained my research. And then the next time I went out and recollected, I actually talked to her a little bit more, and she told me about the wedding she just attended. And then she told me that the reason she loves living where she does, she lives in the middle of Poughkeepsie. So one side of her house overlooks the street and it's a very busy road. And then the other side overlooks the Falcao. And she said she loves looking out the window because it takes her out of the city setting. She loves to escape from reality. In the same section, there was a bridge that I went under twice, and I had met a homeless man. I met a homeless man. So we had a little bit of a conversation, and he said that if I bring him trash bags and gloves, then he would clean up his home a little bit. The stream also went by people's backyards. So I talked to quite a few people just by asking them to use the stream in their backyard, because of course I'm going on their property, so I need their permission. And so it gave me the chance to explain my project, but them also explaining what the Falcao meant to them and how their kids play in it or how they grew up using it. Another, in the city of Poughkeepsie, this man was watering his garden with the Falcao. I also had helpers. I had about 13 people come out with me. Out of those 13 people, 6 of them were non-science majors, and 3 or 4 of them, this was their first time doing research. And every day was completely different because every part of the Falcao stream was completely different. Sometimes I would be high, the water would be high to my chest and my waders would get filled and we'd be swimming. Sometimes it was super easy to walk through. Sometimes it was so silty that it would take 5 hours to get 20 data points. So every day was completely different, but I'm really happy that I was able to expose so many people to my project throughout the summer. And another thing that really made me a little bit sad walking through the Falcao, especially in the city section, was how dirty it was. In my story map, you can kind of, I have some pictures explaining, but I saw about 10 shopping carts and 3 mattresses, tons of trash. There's an AC unit. The Falcao really needs cleaned up. So that's definitely a project that I want to get the City of Poughkeepsie community members involved with, and maybe Marist students. But going through that section 3 times or 4 times really opened my eyes that people use it as their trash can. And finally, I want to give a thank you to the Polgar Fellowship for funding my research and believing in my project. And big thank you to Dr. Zion Kloss, who helped me immensely with everything and is a great mentor. Don Meltz, who's my GIS professor, who taught me everything. Dr. Zarnock, who is further testing my water samples to strengthen my data set. Dr. Kepner, who's providing me with FIB tests. Thank you to all my 13 data collection assistants and Marist College for letting me use their labs. And here's my works cited for all of the information that I use for the background of my project. Does anybody have any questions?
Thanks, Emma. Great job. And yes, I hope you're ready for some questions. We have several. I'm going to start with Marty Rowland. Marty's asking, is there a correlation for non-white residents being renters rather than homeowners?
Oh, yes. Okay. One thing that I forgot to mention in my interactive map, you can tell I didn't add my economic data because I still would like to pinpoint specific areas that are from fecal indicator bacteria rather than just fertilizer and nitrogen. I want to be able to say like, yes, this spot is from FIB or faulty decentralized septic systems before I add the data on the economics side of it. So what was the question again? Residential versus renters?
Yep. Is there a correlation? Did you find a correlation? Or did you look into a correlation? But the idea that right, non-white residents may be more likely to be renters rather than homeowners.
So the Dutchess County tax parcel data that I used to identify spots that residential properties that had the decentralized septic systems on doesn't specify whether or not it's a renter. It does have data on the homeowner and the amount of land, but it doesn't specify whether that resident is a renter or a homeowner. What I was specifically looking at was if their septic system was on a centralized system versus a decentralized system. And yeah, so renting information wasn't on the Dutchess County tax parcel data.
Okay. Do your data suggest that some state legislation is needed to reduce nutrient loading? For example, regulating the density of homes on septic systems?
I think I would be able to answer that after I specifically know if some of those spikes in nitrate are coming from faulty decentralized septic systems, because I might be able to go through those spots and realize that the enterococci count is actually not high or not there at all. So therefore, the spikes in nitrate could actually be from fertilizer use. I know there is a law that just passed in Pennsylvania about specific amounts and specific types of fertilizer used. I'm not exactly sure in New York State if that's also a law that is being looked at or has passed. Specifically, I mean, especially since the Hudson River is experiencing so much excess nitrogen. But that's something that I would potentially look into once I get some of my further tests back.
Okay. And then last question from Marty. Are you getting any pushback from real estate interests who say you are depressing home values?
Do I get any pushback?
Yeah, I mean, you may or may not have engaged with any real estate interest, but if you had that kind of dialogue.
Yeah, I actually have not. So I'm gonna say no.
Okay.
Sorry.
All right. Let's keep moving. There are some more questions. Mark Botten had a question. Aside from the septic fields, do you think there are any other significant point or non-point sources of nitrate that affect the stream?
Yes. So I'm going to share my screen again because there's a very interesting spot. Can you all see the interactive map again?
Yes.
Okay. So one spot that had a lot of dark red, so spikes in nitrogen, was this spot right here. what's interesting is there are no decentralized septic systems upstream, but there is a golf course very close, and there is a cemetery very close. And there are decentralized septic systems downstream. So I think it would be very interesting to understand groundwater flow in this area and what specifically could be causing this nitrogen. I am going to be testing for FIB to see if this nitrogen spike is from possible enterococci. But I guess we'll see in future tests to see what specifically is causing this nitrogen spike.
Okay. Sarah had a question. Was anyone reluctant to let you access their property out of concern that you'd find high nitrates and then they'd be liable for a faulty septic system?
No, no one was reluctant at all, because the way I explained my project was focusing on making the Falcao a healthier stream. I didn't use much jargon when I explained it. I kind of made it very simple for people to understand. Of course, I did explain the faulty septic system stuff, but most people were very excited that I was focusing on the Falcao, especially in the city setting. I had a lot of people say that they would love to be involved in a community cleanup, and that they were happy that I was doing research on the Falcao. There was one experience where me and my friend Nat, we had gotten in this really deep section, and we couldn't touch anymore. So our waders filled up, and this couple came out and asked us what we were doing. They were just watching us like flail around in the water. So we got out and like emptied our waders, and I asked them a little bit about their septic systems. And she had said that she had just replaced her septic system, and that her septic system was very old. So no one was reluctant to let me on their property, no.
Okay, great. And then just a comment from Margie Turin that these are great visualization tools, and a comment from Allison Fitzgerald that she loves the idea of the story map. So you're getting some good feedback there.
Thank you.
And finally, Greg had a question. He said you mentioned fertilizers. I'm wondering if you're able to estimate contributions from surface runoff at all versus the septic. Perhaps any measurements from separated storm pipes that directly discharge surface stormwater runoff into the creek.
It's a very interesting question, and I think that would definitely be able to be done in the city setting where you can identify stormwater pipes easily. When I was walking through, there was quite a bit of stormwater pipes, but I don't believe I would be able to estimate something like that.
Okay, and then just finally, just some more comments on the interactive tools. You really hit the mark. Do we, how do you anticipate the data being used by others moving forward?
Yeah, so I think once I get all of my data into this map, and I'm able to actually identify spots where there are high enterococci counts, the plan is to present this to the Falcao Watershed Coalition. That's something that I've recently been part of. They actually interviewed me about my journey up the Falcao, which was really cool. So I guess working together with researchers and planners and helping them understand spatially that some of these spots might be an issue for decentralized septic systems. I know there's a push for the Falcao Watershed. They just recently made a little information sheet on septic systems, and they've been trying to push out a lot of education on centralized septic systems, because it is up to the homeowners to maintain or replace their septic systems. So I guess I would just present this at various societies, research societies. I know there's the Poughkeepsie planner, urban planner. So this might be something that they would be interested in.
Okay, and one last question for Ray Kepner. It says, great job, Emma. I think they'll definitely be enterococci, but not sure they'll be correlated with nitrate across all sites. But what do you think are the negative environmental impacts of nitrate at the average concentrations that you found?
What are the negative Can you repeat that last part? Sorry.
What do you think are the negative environmental impacts of nitrate at the average concentrations that you found?
The negative at the average concentrations I found. Good question, Dr. Kepner. I mean, I think the negative impacts mostly are ecological. Nitrogen like that is a reflection of the water quality. And in terms of the Hudson River, like having this excess nitrogen is not good for cyanohabs. It's also not good for the water quality of the stream.
Okay, well, great job. I think we're going to move on, and we'll go to our next presenter. Thanks again, Emma.
Thank you.
Okay, our next presentation is from Luana Chakai. Luana's from New Jersey City University, and her presentation is entitled Examining the Interference of Fouling Organisms on Chrysostrea virginica Reproduction and Health. Her supervisor is Allison Fitzgerald, and I should mention that Allison herself was once a Polgar Fellow. It's great to see Polgar Fellows mentoring the next generation of Polgar Fellows. And we've seen that on a number of occasions.
Can you guys hear me okay?
Yes, we can hear you, Luana.
Okay. All right, so I worked with Dr. Fitzgerald over the summer, and the title of my project is Examining the Interference of Fouling Organisms on Chrysostrea virginica Reproduction and Health. Oops, sorry about that. Okay, I'm going to be looking at my notes on the side. So oysters are natural filter feeders. They do so by their natural feeding mechanisms. They can filter large quantities of water at a time. They do provide crucial habitat for other organisms, making it a keystone species, meaning the ecosystem largely depends on the species itself to function. Unfortunately, currently, they're considered functionally extinct, which means that they're not performing the services that they're supposed to in the Hudson River estuary. And they've been replaced by mud-dwelling organisms and some fouling organisms, which usually settle on rocks and hard structures, like the calcium carbonate shells of the oysters. The overpopulation of fouling organisms causes a number of issues, which includes fewer competitions for space. And little is understood about the fouling community, despite the considerable ecological impact. Recently, oysters have shown signs of low reproduction and low spat settlement, meaning we're not finding a lot of them. If you can see on the picture, this right here, these are the spat settlements on the oyster shells, and this is what we'd like to see. And this is the ones that we collected. There's not a lot of spat settlements. And the ones that did have spat settlement, we decided to keep back, and we decided not to take to the lab. For tree restoration success, oysters need to be able to maintain their population on their own. And unfortunately, adequate recovery right now is likely to only to be achieved through human intervention. And so the main goal of the study was aiming to gain more detailed insight on the interaction and the relationships that occur between fouling organisms and the eastern oyster, Chrysostrea virginica. The main hypothesis of the study and what we focused on, it was hypothesized that the increase of fouling community diversity and abundance, it was hypothesized that the increase of fouling community diversity and abundance will result in lower overall condition and less reproductive output in adult oysters. So the first methodology right here were the test sites. So oysters were sampled over 1 to 2 week periods between June and August. And the samples were collected between 2 main sites, that's the Bronx River and Pier 40. It's really important to note that the sites do not have identical conditions. As you can see on the water quality statistics, they're similar, but they're not identical. And that's because we weren't comparing the sites. We wanted to see what we could find at each point and have each site and have two different points of references. As you can see here, the Pier 40 is at the bottom and the Bronx River is up top. Important to know as well right here, this is the Bronx River, the Soundview Park where we took the oysters. Right here, the oysters are kept on open crates. This is where you would see them right here. And at Pier 40, this is the pier, and these oyster crate cages are sealed and closed, and they're suspended right here. So we pull them out, and that's how we sample them. So very different environmental conditions. For species identification, at sites, 20 oysters of similar sizes were collected at random and measured. You can see here with the caliper, pretty standard, and photographed both at the top and the bottom using the photograph device. So I created this device because we needed a standardized way to take pictures of oysters. We were taking 20 pictures of the top and the bottom bivalve. And so we needed a method to take pictures so that when I do the photo analysis, it was completely the same. And so what we did, we took a container, the top of the container, we turned it upside down, became our background. And the top of the container became our bottom. I'm sorry, the bottom became the top and the top became the bottom. We cut up a hole at the bottom and that was the same size as the camera hole. And we measured the phone so that every time I placed it, it was the same standardized picture all throughout. The external fouling organisms were visually analyzed and recorded for species identification. We did that both on the field very quickly because some of the organisms did fall out from touching transportation. We kept them in a Ziploc bag. So for accuracy, we did do that before and then in the lab. 10 of the oysters were taken back to the lab on ice. Condition index and gonad smears were individually assessed for each one. That's what you can see here. Once they were shucked, we did smears of the gonads. And right here, you can see a typical gonad smears underneath the microscope. These are eggs. And so it indicates that this was a female. Oyster tissue and shells were separated and weighed, dried at a 60-degree oven over a 48 to 72-hour period. The dried shells were photographed for internal fouling analysis and identification. And the shell thickness of dried shells were also measured. I measured at the center and at the edges. So it would be right here where it's thickest and at the edge where it indicates that the oysters are growing. So the photographic analysis. So according to NOAA, this is a common way to perform random sampling and percent coverage, which is to take a grid and put it over the picture. We debated a lot on whether to put the grid while we took pictures, but that did not work. So we settled for this, where we found out, I researched a little bit about it. And so the photos were uploaded to a program called Procreate, and a grid was placed over each picture using the same dimensions and pixels throughout. That's the first picture right here. We have the oyster and the pixelated grid was placed over it. A border was drawn around the oyster representing the total surface. This is shown in white right here. So this is what I'm accounting for the total surface areas. The individual fouling organisms were identified and tallied both individually and as a total for each site, which I'll discuss a little bit later, but those were not done on the photographic analysis. Those were done separately, which is what I mentioned before when we did in the field and also in the lab. And then the colonial fouling organisms were identified in the approximate surface area, which it took up, were circled in green. That's what you can see here. So these are all the colonial fouling organisms, which I'll also touch upon a little bit later. The surface area of oysters were calculated by counting how many pixelated squares it took up. So here I did the math and I counted for inside that white border, how much of the pixelated squares there was for the total surface area of the oyster. And the surface area of colonial organisms was calculated by counting how many pixelated squares it took up. So again, the green right here, and I counted how many squares that it took up to see how much that was. And the percent coverage was calculated by dividing the total surface area of the oyster by the surface area taken up by the colonial fouling organisms. So right here was what I mentioned. This is a photo on the field. This is a photo in the lab, and these are the dried oysters. And so the organisms may have fallen off during transport. Therefore, the individual fouling species were initially and quickly recorded and thoroughly examined in the lab to avoid discrepancy and to maintain accuracy all throughout. So condition index. So briefly to explain, condition index is a tool used to estimate the effect of different environmental factors and the effects that they have on oyster tissue. It compares the tissue mass to the whole mass of the oyster. An important rule of thumb, the more gonads, the higher condition index because there's more tissue. That's what we like to see at the beginning of the season where they haven't spawned yet. And the higher index indicates overall good health. Unfortunately, as you can see, there's consistently low condition index throughout. And there is a higher index at the beginning of the season and relatively lower condition index at the end of the season, which is an indication that they did spawn. I performed t-tests, which is used to determine if there's significant difference between the average of two data sets. And in this case, for the Bronx, the first and the second visit, the t-test did not show any significant difference between the two data sets. And for Pier 40, the only significant difference was between the second and the third visit, where the oysters condition index was the greatest. These are the overall Gonad results that we came up with. Just briefly, this is what a good gonad is supposed to look like, and this is what a poor gonad looks like. It's clear, there's not white and milky stuff, and this is what we're looking for. And so this indicates that they haven't spawned, this indicates that they have, and this is a typical gonad smears with the eggs. Overall, there were 6 total females in the Bronx River, 0 males that we could find, and 13 of them were undetermined. In Pier 40, a total of 4 females and a total of 2 males, and 34 of those were undetermined. So we took samples of 20 oysters at Soundview Park at the Bronx, and we took 40 samples at Pier 40. And so majority of those were undetermined. This is an overall review of the species that we did see. Relatively the same species all throughout, with very few that are different between the sites. Algae, as more prominent, was the algae on the Bronx River, and anemones for the Pier 40. They both had things like boring sponge and lacy bryozoans. And important to note that the ones that I highlighted in blue, they're not complete fouling organisms, but they're considered quote unquote passerbys, where they can move on from oyster to oyster. They settle on a hard surface like barnacles and slipper snails, but they can also move on to other oysters and not stay on that particular shell forever, like a lacy bryozoan would. However, I counted for the entire species richness because there's still competition for resources in space. And some of these work from the outside in and some from the inside out. So we accounted for fouling organisms on the outside, such as mussels and anemones, and from the inside in, such as bloodworms, clamworms, where they burrow into the shell. And so the integrity of the shell is compromised from the inside out. A lot of the times when I was shucking these oysters, they did break apart because of the sponge damage and the tube worms and the bloodworms that burrowed inside. So some of the results that we were able to come up with these are the total species abundance and frequency. And so right here, you can see that other than the mud snails and oyster drills, so these are the mud snails and these are the mud snails eggs, and these are the oyster drill eggs. There are no significant differences between the other fouling organisms. And that's because these two, they were eggs at the beginning of the season. So at the first visit, right here indicated in blue, and at the second visit, they hatched. So they weren't part of that, they weren't settling on the oysters, but they were rather already organisms that were not settled, and they were not eggs anymore. So here I accounted for the average % coverage of colonial organisms. Again, bringing back that these are the colonial organisms, mostly algae in the Bronx River, including boring sponge, which we did see a lot too, and lacy bryozoans. And so right here, you can see that the first one, the average was a little lower than the second visit. And when performing a t-test, it showed no significant difference between the 2 data sets and the 2 samples. This is accounting for what I did at the photo analysis. Right here is what the lacy bryozoan, for example, is taking up. These are algae, is taking up on the overall coverage of the shell. And then we count for how many, and that's the average % coverage. So the results that we came up with for Pier 40 and for the abundance of organisms. So overall, there was a decrease in species abundance throughout the summer. So from the beginning of the season to the end of the season, from the first to the last visit, with the exception of anemones, which are right here, are the first ones, tube worms right here, barnacles and sea squirts. There was an increase higher between the second and the third visit. And that's because there's a correlation between fouling spawning and warmer waters. And so as the water increases and the water warms up throughout the summer, there's an increase and there's a burst in these fouling organisms. And that's an indication of spawning. So for the average percent coverage of colonial organisms, again, here, there's a significant difference between the first and the second comparison to the second and the third visit, as again, shown by the t-test. And here we were considering boring sponge and lacy bryozoans as the colonial organisms. And again, here I shows these are lacy bryozoans and some of the boring sponge damage that was done to the top bivalve of this oyster. So I came up with a system. So these are, I categorized the oysters between low, medium, and high infestation. And so in order to do that, we needed a standardized way to do so. And so I came up with this graph and this way to categorize them. And so the way that I came up with was, it was a positive correlation. So as percent coverage increased, so did the species richness. And so what I did was the percent coverage was divided into 3 low, medium, and high categories. 100% divided by 3 categories, which stood up at 1 to 33%, 34 to 66%, and 67 to 100%. The low, so species richness and species abundance, on average, each oyster had a minimum of 5 species per site and a total of 10 plus species per site. So 10 species divided by 3, low would be 1 to 3, medium 4 to 6, and high would be 7 and above. So important to note as well that severe, we didn't find any severe, and I'll explain in a little bit. And that's because a lot of these shells were highly infested on the top bivalve or the bottom, but not the other, its counteractive part. And that only accounts for 50%. And so what the most that we did find was heavy fouling, but most of them stood on this area right here, which was the low. So right here, I categorized them into test sites. So the number on the graph indicates the abundance of oysters in the category per date. And as you can see, majority of them are at the bottom. We had 11 on the heavy fouling, but majority stayed within the low percent coverage with a range between low, medium, and high species richness, with the highest species richness and lowest percent coverage overall being the most abundant and the most frequent. So here's just an example of what that means. So on this one, these are all the bronx shells from our second visit. Right here is the low. There's very minimal fouling organisms, so very low species richness with low percent coverage. There's a few sponge damages, boring sponge damage. And right here is the medium species richness. So there are a couple of other organisms and the algae, which would account for percent coverage as well. And this is a high species richness. So right here are tunicates at the bottom right here, which would account for percent coverage, and other organisms like the mussnail eggs. And right here is what an example of a heavy fouling. So right here on this side at the top bivalve, it's completely fouled by the algae, but at the bottom, it's relatively clear. So this is only accounts for 50%, which I did mention before, which is why we wouldn't reach the severity. And so in conclusion, the conclusions that we made thus far, and I'd like to note that there's an in-depth statistics is still pending. We're still working on that. There are no differences in the abundance of fouling organisms at Soundview Park over the summer. There's a slight decrease in abundance of individual organisms over time in Pier 40. The percent coverage decreased slightly at Pier 40. And the biggest significant difference between Colonial percentage coverage and Pier 40 site between the first visit and then in comparison to the second and third visit, which the t-sets indicated were the most significant. Linking of condition index and gonads of fouling intensity is still pending. However, more females were seen at Bronx, and the drop in condition index indicates spawning, which was what we wanted to see. Overall, fouling intensity was low at each site, with most infection due to tunicate and sponge on the shell. Species richness was high, but a lot due to mobile fauna that live on the reef. And there was a medium, so overall, there was a medium percent coverage. That's what we saw. And there were medium to high species abundance. I'm sorry, there was low percent coverage and there was medium to high species abundance, which is why we saw most of them at the bottom at the green indicated on the graph. So I'd like to thank the River Foundation, the Hudson River Foundation for funding my project. A special thanks to Dr. Fitzgerald for going above and beyond and being an amazing mentor. A special thanks to the Hudson River Park Trust at Pier 40, Carrie as well for helping us out and for always being a great helping hand, and for site access and field assistance. And thank you to Kayla, Hui, and Dr. Carroll for lab assistance all throughout the summer. Thank you.
Okay, thank you, Luana. Great job. We have a question from Judy Weiss. And Judy asks, oyster spat typically settle on shells of adults, so shouldn't the oysters themselves be considered fouling organisms as well? It looks like you didn't find any, or did you not count them?
Can you repeat the question? I'm sorry.
Judy points out that oyster spat settle on the shells of the adults. So shouldn't the oysters themselves, shouldn't they be also considered fouling organisms? Did you find any spat on the shells, or did you not count them as fouling organisms?
So we did find spat settlement on the shells of the oysters. We did not consider them fouling organisms because that's what we are trying to see. The fouling organisms are taking up the space for these spat settlements to settle on, and that's not what we wanted to see. And so we did see spat settlement scars on the ones that we did collect and bring to the lab, but the ones that did settle and were alive, we chose not to bring those because we want them to have a chance to reproduce. So no, they were not considered fouling organisms. And the other fouling organisms that we did consider were encrusting crustaceans and cnidarians and mollusks.
Okay, great. Thanks very much. I think that's it for the questions. And so we'll move on to our next presenter. Oh, hold on. There was one more question coming in. Let's see. Mark Botten asks, did you see any relationship between fouling organism species diversity and the size of the host oyster?
There actually was. I briefly mentioned to Dr. Fitzgerald. So the Soundview oysters at the Bronx, they're much older and they're much bigger. So majority of the oysters on the heavy fouling on that graph was under Soundview, and that's because the size of them, they have more, they're older, so they have more space and more time that these settling organisms and fouling organisms were able to take over. The Soundview, I'm sorry, the Pier 40, they were much smaller and they were much younger, and so they weren't fouled. And it's also important to note that they were in cages, and so a lot of the times when we pulled out the cages, a lot of the fouling organisms were on the cages itself. And so at the Bronx River, they're open and they're out in the open, so it's easier for the fouling organisms to get to them.
Okay, actually we have a couple more questions. There's one from Hoyt. Could it be possible that the missing presence of extremely heavy coverage shells are a result of survivorship bias? The ones with heavy coverage of fouling organisms would have died possibly.
That's actually possible. That's a great question, and that's an actual possibility. Dr. Fitzgerald did go to another site with one of her colleagues, and the amount of fouling and the different types of fouling organisms were a lot more prominent, especially the boring sponges where it was kind of coated. And so it is possible that the oysters have died before, before we could get to them. And that also can be an indicative of the condition index, which are already low. So their health are not exactly probable and they're not exactly high. So it could mean that they're dying before we even got to sample them. Yes.
Okay, great. And one final question from Marcia. Can you comment on how predator organisms might be different in a natural oyster reef?
Predator organisms might, I'm not sure what you mean by that question.
Okay, Marcia, if you want to retype that in the chat, maybe she can log into the chat later and answer that, or you can maybe talk offline.
I'm not sure, but Chester had his hand up. I'm not sure if you had a question.
Chester, let's see. I don't see Chester.
Yeah, I'm here. I'm here, Dave.
Okay.
I did have my hand up. Luana, good job. Lots of really interesting data there. You kind of hit on my question when you were responding to somebody else's chat question. I'm really interested in understanding how some of the percent coverage data might relate to condition index, body size, and so on. So you mentioned that you were going to be working on that. So I look forward to seeing that coming up. Yeah. Thanks. Good job.
Thank you.
Okay. I think Marcia just clarified her question, is that your perspective seems to be that the fouling organisms are limiting the oysters from thriving. Would you
Correct.
agree with
So
that?
there's a difference between predatory organisms and fouling organisms. Fouling organisms generally are considered the ones that settle on a substrate. Predatory are a little different. We did not account for predatory because that can account for a number of other organisms that we wouldn't necessarily be able to account for. And so fouling organisms can be predatory, like a drill snail, for example. They're fouling because they damage the integrity of the shell, but they're also using the shell as a resource. And so that's what we try to look at, how these behaviors and how these fouling organisms trying to survive, they alter and they affect the overall fecundity of the oyster.
Okay. All right, and then Ray Kepner just has a comment that you might want to look at using a non-parametric test versus the conventional t-test to look at your data, but we know this is a work in progress, and I'm sure you're going to run a whole lot of exploratory tests on your data. So, but
I appreciate it. Thank you.
We do appreciate the feedback, and I do want to make that comment before we go to the next presenter, that we really appreciate all this Q&A and this participation from the audience. This is really helpful to the students, and the fellows are doing a great job in responding to these questions. They make it look easy. Okay, so we'll let you go there. We'll bring up our next presentation. Okay, next up is Max Brown. Access from Queens College, CUNY, and his presentation is entitled Scaling Phytoplankton Assemblage Responses to Nutrient Quality Following COVID-19 Stay-at-Home Orders from the East River to Western Long Island Sound. And Max's advisor is Diane Greenfield.
Hey, thanks so much. So my name's Max, and I'm a second-year master's student at Queens College. My project is slightly different now. It's evolved a little bit. So we're calling it Comparing Phytoplankton Assemblage Responses to Nitrogen Form Following COVID-19 Stay-at-Home Orders from the East River to the Long Island Sound. So now, instead of scaling, we're kind of thinking of more of a comparison. So this is all part of a larger project that's going on in the Greenfield lab, trying to understand how human behavioral changes associated with the COVID-19 pandemic, so stay-at-home orders, have altered the biochemistry of NYC waterways. More specifically, I'm looking at how the forms and concentrations of nitrogen have been altered until around June 2021, which is when the disaster emergency was sort of declared over because of COVID-19. And so I want to see if there are measurable changes due to COVID-19, and the real question is, is degree of changes things that phytoplankton are going to respond to, and that's going to be based on field observations and experiments. And so this summer, I was really fortunate to be able to expand some of my work into the Upper East River, and particularly at Frog's Neck, which demarcates the boundary between the East River and the Long Island Sound. So I'm going to be focusing on that data set. So this is just some introduction. Basically, New York City coastal waterways get a huge amount of nutrient pollution in the form of nitrogen. Sewage is the predominant contributor of nitrogen via point source pollution, and this can be treated or raw sewage when you have a sewage overflow event. Dry and wet deposition of nitrate and nitrite is also going to be about like 15 to 40% of the total river load in the Northeast. And so this is a problem because nitrogen is a limiting nutrient for phytoplankton, and so they grow rapidly in response to these additions. However, in most of New York Harbor, this isn't a problem because you have rapid flushing time. So the East River is a light limited system because it's very turbid. There's a lot of sediment. And so it doesn't experience that many blooms. However, it does exchange water with the Long Island Sound, which is not light limited. And it doesn't have the same rapid flushing and has a higher residence time. So the nitrogen load in that system, the Long Island Sound, has been associated with seasonal hypoxia, which is low oxygen levels, which starts at the Western Long Island Sound and then moves east. and it's also been implicated in causing some harmful algal blooms. So different nitrogen forms are available at varied concentrations due to both natural and anthropogenic sources. Coastal phytoplankton communities have come to respond differently to each of these inputs. It's been shown through some experiments that nitrogen additions, especially organic nitrogen, produces elevated biomass near urban areas. So you basically get a localized effect due to the particular ensemble of urban nutrient inputs. What we also know is that there's sort of differences in the way that individual groupings or like taxa, phytoplankton, uptake nitrogen. And I'm generally going to be focusing on reduced forms of nitrogen, oxidized forms of nitrogen, and organic forms of nitrogen. So for example, the reduced forms like ammonium are more rapidly used by certain dinoflagellates. An example which is shown here on the right, which is Protocentrum. And then cyanobacteria also really uptake reduced and rather quickly. Oxidized nitrogen, so like nitrate and nitrite that you get from cars, is more rapidly used for growth by diatoms. And then you have an example here on the left in that image, which is the chain forming skeletonema. And then, so the larger organic molecules actually tend to be uptaken by dinoflagellates or other flagellated species through a process called osmotrophy, where they're actually able to diffuse the large molecule like an amino acid through the cell membrane. So this is all kind of getting into the ecology behind the differences in nutrient sources. And this is important because if you have the right environmental conditions, and it's a certain type of phytoplankton, you can end up with a harmful bloom. So there's been, in the past decade or two, there's been a bunch of harmful blooms of particularly dinoflagellate species in the Long Island Sound. So as I mentioned before, the disaster emergency was in place from March 2020 to June 2021 due to COVID-19. So this is kind of the time frame to look at the anthropogenic impacts. So the question really is, how did COVID-19 impact the forms and concentrations of nitrogen? And so kind of taking some observations and then kind of thinking about how that would impact the local water system. We know that stay-in-place orders contributed to a 25% atmospheric nitrate and nitrite decrease in urban counties, and that's due to decreases in automobile transportation. And we also know that the percentage of employees working from home was increased from around 8% in February to around 35% in May 2020. So that's probably going to impact where the wastewater inputs. This figure actually shows you how you kind of have like a skew between what you normally have with the amount of nitrate in the air. So there's some scientific rationale for understanding that there are going to be different inputs into the system. And so my study goal is really to understand, how these behavioral changes have influenced ecological and water quality parameters in surrounding New York waters. So the ecological is the phytoplankton community, and the water quality are these nitrogen levels. And so with the support of the Polgar Fellowship, this was able to be expanded into the East River. And so the first objective to kind of achieve this goal is sort of observational. And that's really just documenting, bookkeeping, understanding what species are where, what's their biomass in terms of chlorophyll A, and then what sort of nutrient forms are available in the Western Long Island Sound and the East River. And this was basically a year's worth of gathering data. And I started my role in this around last fall. And the second objective is experimental. And in this experimental part, we actually want to try to figure out the mechanisms behind sort of the relative ratios and relative concentrations of nitrogen species and what's going on with the phytoplankton that you're seeing. And so in that objective, we want to see how these phytoplankton are responding to nitrogen additions at two different locations. One of them is going to be in the East River, and one of them going to be more in the Long Island Sound. And these were executed during summer 2021. I'm going to spend most of the time talking about the one a little bit closer to the Hudson River, so the Throgs Neck bioassay we did in July. So this map kind of breaks down where the data in the study is coming from. So the observational data is from a variety of sources. There's sampling every 2 weeks in the Western Long Island Sound with the Connecticut Department of Energy and Environmental Protection at 2 depths, the subsurface and the subchlorophyll maximum. Those are the red lettered sites in this figure. At all these sites, this is accompanied by small boat surveys, which are done on occasion when there's something interesting. And then there's regular shore sampling monthly, which are the yellow locations. I added a location this summer at the Sound View Pier to kind of fill in some of the gaps for connecting this system kind of to the Hudson River. So at all these sites, physical water quality parameters are recorded, and then we're transferring water in triplicate to the lab. And then the bioassays, which are sort of mechanistically trying to understand what's going on. We're at 2 different ends on this map, and these are the kind of aqua blue stars. This is where Throgs Neck is right here, and so that's really capturing sort of like the East River coming into the Long Island Sound. And then the Saugatuck River, which is actually a predominantly the land coverage there is rural. So here you have like a nice system for understanding sort of a rural freshwater input, and then here you have more of a sort of urban freshwater input, and that's kind of the design that we had in mind, thinking in terms of a bigger picture. So then just some more background on how we're actually measuring this data. From each triplicate water sample that we take, we're interested in 3 main things: the nutrient concentration, the chlorophyll concentration, and then the phytoplankton community. So for nutrients, we aliquot all the water into scintillation vials for analysis of total nitrogen and phosphate, dissolved nitrogen and phosphate, ammonia, nitrate and nitrite, and silicate. For chlorophyll, we filter into 3 size fractions to analyze chlorophyll contributions of 2 different phytoplankton, so total and then less than 20 microns, and then 1 picoplankton size, which is less than 5 microns. And then for the counts, we preserve samples, again in triplicate, in the 5% solution of Lugol's iodine, and then they're enumerated on a Sedgwick chamber, which you put 1 ml of your liquid on there, and you calculate the sort of concentration constant, and then you basically count 1,000 squares to quantify the number of each different type of species in the sample. And then you know how much is your concentration, so then you calculate what you would expect your concentration per mil. So those are kind of the methods. Now I'm going to switch to talk about sort of the bioassay mostly. And to kind of give some context for that, and to kind of show you guys what the impact of COVID looks like, I have these sort of the summer of that values. So here's some historical data that gives us a peek at the biogeochemistry of the water moving through Throgs Neck. Here I'm plotting New York Department of Environmental Protection data at E8, which is right at Throgs Neck in the middle of the channel. And this is from 2010 to 2019. So these are seasonal means for June, July, and August. I'm showing 2010 to 2019 because nitrogen concentrations over time have been steadily decreasing. So we don't want to see a lower value because of COVID than the mean. And we think that that's something significant, but we're actually comparing that to super eutrophied waters from mid-1950 or, so the error bars are 1 standard deviation above the mean in either direction on this figure. So basically what we see at Throgs Neck are typically high nitrogen levels, like most of NYC waterways. However, there do appear to be some small seasonal trends. I think that the nitrate and nitrite concentrations tend to increase into the summer. And the big error bar that you see for the green nitrate in August is because sometimes you get extremely high values towards the end of the summer. This is also coupled with an increase in total phosphorus levels. I think ammonia, it's kind of a bit unclear what's actually going on. So these values are kind of the baseline for what we're going to think about the impacts of COVID-19, and then how we're going to design, how we designed the bioassay experiments. So here I plotted data points from the summer of 2020, which was the first COVID summer, to give an idea of how that COVID fits into the historical range and to give an idea about the perturbations, right? What jumps out to me is that it really looks like in June 2020, nitrite and nitrite were kind of below the normal range for values. It's hard to really say if big ammonia peak in August. So I think from here to say, from here, like what my takeaway is that the changes due to COVID-19 are likely to be very subtle, especially at, probably in this area around Throgs Neck and the Upper East River. So these observational data allowed us to understand the context for our experimental design. So in order to determine the specific drivers of phytoplankton assemblage shifts, we planned a number of in situ, in location bioassays to assess how reduced, oxidized, and organic forms of nitrogen impacted phytoplankton. And so again, I'm going to focus on this Throgs Neck bioassay, and the plan was to do this twice, one before hypoxia and one during. But there are some problems with that I'm going to explain in a little bit. So basically for these experiments, we collected water on site, and then ammonium, nitrate, and urea were added as 20 micromolar concentration spikes to clear 2-liter Nalgene bottles in replicates of 3. 20 micromolar was chosen because it's a reasonable increase based on the historical data we just looked at. So it has some sort of ecological relevance to the system. We also added phosphorus at 1.25 micromolars to prevent co-limitation. And then finally, we added 2 additional treatments, one with all the nutrients at a total of 20 micromolar, and then one with no additions. So the bottles were placed in a mesh bag that blocks 60 to 70% of sunlight to keep the phytoplankton at maximum productivity. This bag floated at the subsurface, or just really at the surface, where phytoplankton were exposed to natural light cycles and temperatures, obviously with the caveat that there is some filtration of the UV. And then we subsampled at T0, T24, and T72 hours to get a full idea of how nutrient concentrations and phytoplankton are changing over time. We brought the subsamples to the lab to filter for ammonium, nitrate, nitrite, organic, and phosphated silicate, and then we measured chlorophyll. So I'm going to share those data in green with you. So this is kind of the location at Throgs Neck, which is a southwest facing pier. There's the white stone bridge in the background. Here's the bag that's holding all the bottles together. And you can see the zip ties stitched together to make sure the experiment didn't float away. And then here's all the bottles upon return to the lab at T = 72. If you look carefully, you'll notice that the colors of the water were actually different. And so the clear one second from left is the control treatment next to the all treatment. And you can see that the nutrient additions had an impact on appearance. So at time 0, before any nutrient additions, the phytoplankton biomass, which was measured by chlorophyll A, which is here on the y-axis, was predominantly in the less than 20 micron size fraction. So these are mostly nanoplankton. And what this means is that cyanobacteria, which tend to be smaller, so like less than 5 microns, were really a small portion of the total chlorophyll A biomass in this assemblage. When I counted this assemblage using the Septic rafter chambers, there was a predominance of diatoms, which is to be expected, I think, in this location. Notably, Thalassiosira was around 1,700 cells per mil, and Skeletonema was around 300 cells per mil. So again, these were kind of very common groups. So here's what happened after 72 hours. And then looking right now just at the control, the mean chlorophyll A biomass for the control group increased from 8 to 75 milligrams per liter, and the counts more than doubled. So Thalassiosira, which is a chain-forming diatom, went from 1,700 to around 9,100, and Skeletonema increased from 283 to 938 cells per mil. So this alone, just with the control group, kind of tells us that the conditions of the bioassay, being enclosed with a constant 30 to 40% irradiance at the surface, are promoting phytoplankton growth. So then, we take a look at these other treatments, and as we were analyzing the data from these experiments, counting the cells, measuring chlorophyll, measuring nutrient concentrations, we noticed something wasn't quite right. there was a big problem actually with this experiment. So the problem was that the nutrient concentrations were increased by way more than 20 micromolars, something around like 700 to 800 micromolars. At this point, we realized that there was probably an error in the calculation of the design. Like we can talk about this data, but a large increase in concentration isn't going to tell us much about how the phytoplankton would respond towards real anthropogenic perturbations in the coastal urban environment. But from what we're looking at here, you can see that there is kind of a ubiquitous response above the control treatment. And then when I started actually doing the counts in the Sedgwick chambers, I found that nearly every grouping increased. So the counts for Thalassiosira were 12,000 versus 9,000 at 72 hours, and Skeletoniema was about 3,400 versus 1,000. And so it wasn't just these chain-forming diatoms that were increasing. I found similar increases in dinoflagellates, cryptophytes, euglenophyites. Everything was basically enjoying these massive amounts of nutrients. So that takes us to next steps. And really, I want to repeat this experiment in the next week or two. We found the error in the calculations, and we're currently in the process of running some tests to make sure that we have it right before we get back into the field. I didn't focus on it for this talk, but I've been sampling at Soundview Pier, similar to the talk just prior to me in the Bronx. And this is to the west of Throgs Neck. And this is to kind of contextualize sort of the results I'm seeing in terms of the local phytoplankton ecology. And interestingly, in the first half of the month, so August 4th to August 17th, the dominant species shifted. In the beginning of the month, it was mostly diatoms, but now we're seeing a lot more flagellates. And so the point there is that depending on when we kind of conduct this bioassay, we're going to have a different starting assemblage, and that's going to tell us about a different kind of group of species. And so that's why we wanted to do two, one in the beginning and one kind of at the end. But we're going to kind of do the best we can given this. And so basically, ultimately, we want to take both the shore observations and the experimental bioassay data together and put them through the framework of available nutrient concentrations. And then by doing this, we want to try and kind of piece together how some of the very subtle changes due to COVID-19 may have impacted local phytoplankton communities. I really want to thank everybody in the Greenfield lab for helping so much, particularly our research assistant, Maria Paula, and Dr. Greenfield herself. And a special thank you to Dr. Katerina Panzeka at SUNY Maritime for helping me have access to the pretty perfectly located pier at Throgs Neck. And so happy to answer any questions, and thanks for your time.
Okay, great job, Max. Do we have any questions? I don't see any questions for Max. Oh, here comes one from Ray Kepner. Ray was just making a comment. Great work. Acknowledges how tedious the counting time is with the Sedgwick rafter cells. And he has a question: are there any concerns about CO2 limitation of growth due to sample? I'm not sure what you.
Maybe oxygen. Well,
bioassays, yeah.
The CO2 and that's actually a really good point. So the way that we did it was in the design at 24 hours, we're going to open up these bottles to take a subsample. So I'm assuming, I think what we were thinking is when we open up those bottles at that 24-hour mark, we're then kind of letting in new air and kind of replacing the old air. So maybe you're going to get some ambient CO2 and some oxygen that's going to come back into the bottle. But we didn't, they're growing a lot. Like if we go back to the slide, it just becomes they really like what's going on in these bottles. So I'm not too worried about limitation, I guess.
Okay. All right, I think we're ready to go for a 10-minute break, and then we're going to come back the final two presentations. Helena, did you have any instructions for the participants for the break?
Sure. We'll come back at 12:15 But before we go, I want to say great job to the fellows. It's their wonderful presentations. And I also wanted to point out that after the initial introductions this morning, we had several advisors join us. So I wanted to acknowledge them and let you know they're here. Rebecca Hauser from New York State DEC is here. Raymond Kepner from Marist College, who's been asking lots of questions, and Diane Greenfield from Queens College, CUNY. So we'll see everyone at 12:15. There we go. Okay, everyone, it is time to begin our final session. And our first speaker is Ashley Smith Krudner from Queens College, CUNY. And her project is investigating the influence of sediment resuspension on the bacterial water quality of the Harlem River. And her supervisor is Gregory O'Mullen. So Ashley, go ahead. Ashley, I think you're still on mute.
Sorry, thank you. So good afternoon, everyone. My name is Ashley Smith Krudner, and my project is investigating the influence of sediment resuspension on the bacterial water quality of the Harlem River. Just to give a little bit of background about myself, I'm broadly interested in the effects of an increasing human population and urbanization on our local water quality. With the continuously growing human population and aging or outdated infrastructure, this often leads to sewage contamination in our local waterways. With sewage contamination comes fecal contamination, which is indicated to us by the presence of fecal indicator bacteria, also known as FIB for short, in the water. Though FIB don't pose a threat on their own, they do indicate that there are possible health risks like fecal pathogens, which cause disease in the water. According to Young et al., a prior Polgar Fellow, fecal concentration is often attributed to wet weather discharges and sewage contamination. And despite having thought to understand this, there are other drivers and complexities that are now being considered. So one of those complexities is particle association. Particle association, according to Sutter et al., approximately 50% of FIB are particle associated. Looking at the figure to the right, these particle associated fecal indicated bacteria will eventually sink and be buried in a sediment bed. This sinking flux matters to change in fecal indicator bacteria concentration over time, according to Myers et al. So after sinking and being buried in the sediment bed, the sediment ends up being an environmental reservoir, protecting FIB from turbulence, sunlight, high temperatures, and predation. In other words, sediment provides a favorable environment that increases FIB lifespan outside of the gut. Prior studies have shown that sediment FIB concentrations in the Hudson are often orders of magnitude greater than that of the water. This comparison was done on a per mass basis where a milliliter of water, which is equivalent to 1 gram, is compared to a gram of sediment. This is confirmation that sediment can act as a reservoir for FIB. Particle-associated FIB do not remain in this reservoir and can also be removed from it through resuspension, as seen in the figure to the right. Resuspended particles are reintroduced to the water column, and this includes those particle-associated associated fecal indicator bacteria. This tells us that resuspension of sediment has the potential to influence FIB levels in the water column, even during dry weather. For example, a simple resuspension experiment conducted on the Hudson shoreline, in which samples were collected before and after the sampler entered the water, showed that entering the water can impact FIB concentration through resuspension at a level that is relevant to acceptable water quality conditions. The graph on this slide is showing the change in FIB concentration before and after the sampler entered the water for that experiment. This is shown for 10 experimental resuspension trials. And in 60% of these trials, the change in FIB concentration alone exceeds the beach action value, or BAV. This tells us that shoreline disturbance of sediment can cause unacceptable FIB levels even in dry weather. This project took place in shallow water near the disturbance, but what if we apply this to other larger scale ecosystem disturbances, such as boat wakes? This question led me to the overall objective of my project, which is to determine if boat wake disturbances of sediment can significantly increase FIB levels in Harlem River user locations. Included in my approach to this was collecting FIB and total suspended solids samples in parallel with USGS continuous water monitoring data on the Harlem River. Turbidity and wave height, or surface elevation, were the two parameters we focused on from that data. I also characterized FIB abundance in sediment along a park waterfront along a park waterfront access dock adjacent to USGS sensors. Lastly, I collected water samples before and after large boats had passed, which is the focus of my project. And these samples were analyzed for both total suspended solids and FIB. So what inspired this approach is that recent studies by the USGS conducted on the Harlem River have demonstrated that there is a correlation between sediment resuspension and elevated FIB concentration. Currently, the USGS is studying the water quality of the Harlem River near public access points at 2 sites, including Mill Pond Park and Roberto Clemente State Park, which is the site I focused on in my project. This past spring and summer, I assisted USGS in deploying continuous sensors for turbidity and wave energy, and I also collected weekly FIB and total suspended solid data. The image to the right is showing one of these monitoring stations, and this is at Roberto Clemente State Park, and this is right along the Harlem River. The background data collected by the USGS monitoring station showed that TSS and turbidity were positively correlated, with around 85% of the data being explained. FIB concentration and TSS were also positively correlated, but with only around 17% of the data being explained, telling us that total suspended solids is not the main driver. This now brings me to my experimental design. This image on the left is an overhead view of my sampling locations at Roberto Clemente State Park. Samples were collected at the USGS monitoring station along the Harlem River at sensor depth. Samples were also taken from the park dock that is across from the monitoring station. And I also have labeled here the cove in which the resuspension event is occurring. This video to the right that I'm going to show is an example of a resuspension event as a large boat is passing by the cove. This is only one example of what a resuspension event can look like, as it can vary depending on environmental conditions. So before collecting samples before and after the resuspension event I have just shown, my first step was to characterize sediment FIB levels around the dock at Roberto Clemente State Park. I took 4 sediment samples around the dock, and the resulting data showed that FIB concentration was within the range observed in a study by O'Mullen et al. With more extensive sampling at Flushing Bay, another CSO impacted waterway. That study took place at 2 sites at Flushing Bay, which were within the same vicinity, but they varied a little bit in the sediment bed in terms of one of them was more muddy and the other was a little bit more sandy, but there was no significant difference found among these 3 sites. So when I compared the FIB concentration in those collected sediment to the park to undisturbed dry weather water samples from the water column, the FIB levels in the sediment were found to be orders of magnitude higher than in the water. I found no significant difference in any of the paired groups of the paired samples that I took, and this confirms that the sediment is an environmental reservoir, and it is reasonable for us to consider that resuspension could have an impact on the Harlem River water quality. So next, I used the USGS continuous monitoring data from the sensor at Roberto Clemente State Park to determine if there was a change in turbidity after the boat wake or the boat went by. The 4 graphs shown here show 4 different boat wake disturbance events with varying degrees of response to each event, but there's a similar trend observed. In each one of the graphs, there is a red arrow that indicates the passing of the boat. As the boat passes, you're seeing an increase in wave height, which is indicated by this blue line here. You're seeing an increase in wave height that continues even after the boat has passed by. So it continues, as you can see after this red arrow, you can see that the wave height is still increasing. The orange data is representing turbidity, and after the red arrow, which is after the boat has passed by, we're seeing that along with that increase in surface elevation or wave height, we're also seeing a increase in turbidity. Now, for all of the events, the response to the event in terms of an increase in turbidity, it's not always at the same time, it's not always as fast, there might be some delay, but we are seeing generally a trend of the event occurs, wave height increases, turbidity also increases. And this is telling us that resuspension is immediately occurring from the sedimented cove that is adjacent to the dock. So here are some visual representations of the before and after of the boat wake event. And this is the dock where I take, this is one of the sites at the dock where I take one of my samples at, and then the area from which the picture is being taken is right above the USGS monitoring station, which is the site of the other site where I take the other set of samples. And as you can see here, there's obvious change in turbidity. And here is another visual representation, but this time we're looking at the cove where the event is originating and where the plume is originating. And we can see here that the plume, and in the image to the right, that the plume is pretty patchy. And we observe that in each event, the plume is different and its patchy nature is always there. So we might collect different areas of the plume, and it might take a while to reach the sensor, it might not reach the sensor. And here you can see that there is also an increase in turbidity in these 2 pairs of before and after samples taken at both sites before and after the event. So now we get to the collected data. Analysis of the data showed that TSS values increased significantly after the boat disturbance in both paired before and after samples in Roberto Clemente State Park dock samples. But unfortunately, when it came to FIB, we did not observe with the current level of sampling that we conducted that FIB levels significantly increased after the boat disturbance in the matched before and after pairs. So overall in the data, we saw that sediment in the area of the dock had an average FIB level of 665 mpm per gram, and TSS in our samples ranged from -34 to positive 336 milligrams per liter. This being the case, we expected that TSS increases in our data would only account for an increase in FIB concentration ranging from 0 to 28 mpm per 100 milliliters. But surprisingly, we observed a FIB concentration change of up to 496 mpm per 100 milliliters. And looking at our data more closely, we feel that this might be due to the patchy nature of the turbidity plume and that the impact is most likely greater in the areas that are closer to the shore. So in conclusion, we found that sediment at Roberto Clemente State Park has high levels of fecal indicator bacteria when compared to undisturbed water from the water column. These levels are higher. We also found that FIB concentration in the Harlem River during dry weather can actually vary and displays concentrations that are of a concern for water quality management and public exposure. We also found that boat wakes significantly increased TSS as expected, but do not have the expected level of impact on FIB concentration, though this is likely due to our current level of boat wake sampling not capturing a strong fecal indicator bacteria sample or signal. So my next steps in this project will be taking more before and after samples, further examining the relationships between USGS sensor data in the full data set, and using sensor data to predict boat wake disturbances. We also have previously collected DNA samples from sediment and water samples that we plan to do DNA analysis on if FIB are found to increase significantly with more sampling. And lastly, we are considering expanding the project to other similar locations like Mill Pond Park. Before I end my presentation, I would like to say thank you to all my project collaborators at Queens College, the USGS, and the IEC. And finally, I would like to give a big thank you to the Hudson River Foundation and the Polgar community for giving me this opportunity, this experience, and enabling me to further develop my project, which will help me with my own career path down the line. So thank you, and I welcome any questions.
Great, that was terrific. Very, very interesting presentation and very interesting work. So let's see, from Ray Kepner. Great job, Ashley, love this work. Method questions. Using IDEX for enterococci, question mark.
Yes.
Any bacterial extraction procedure, for example, sonication, applied to sediments, or are sediments just diluted and turbid water added to the quantitrays?
So in terms of extracting sediment from the DNA from the sediment, we added a chemical that helps to pull off the fecal indicator bacteria from the sediment, and then we did sonicate it for around 30 minutes at a high speed, and then after that we let the sediment settle, and we filtered out around, if I remember correctly, about 10 milliliters of that liquid, and then after that we took around, I think if I remember correctly, we took those 10 milliliters and we incubated that using the IDEX procedure.
Okay, in the Q&A from Marty Rowland. Should the sediment be vacuumed from the river bottoms to control pollutants? If so, how do you limit impacts on protected organisms there? That's a loaded question.
So I don't think we would vacuum the sediment. That'd be a lot of work and probably cost a lot of money. And I don't know. And in my project, I only sampled the top layer of that sediment. So I don't know the concentrations in the lower, in the deeper sediment, but I wouldn't, I personally, I don't know about, I guess, you're talking about dredging the sediment from the area. I wouldn't do that. Sounds, I don't really know, but I personally wouldn't do it. It just sounds very disruptive and very costly.
Okay. And from Chester Zarnock, nice job, Ashley. And the question, did the magnitude of resuspension change with tide level? Low tide equals more resuspension?
Yes, so low tide definitely equals more resuspension. At high tide, the vent, sometimes the change in turbidity was barely noticeable, and also sometimes the I guess the wave action or wave energy didn't really seem to impact the cove. It didn't really seem to reach it, and we also have the dock, which actually blocks some of that action in certain areas of the cove. So that also had an impact on the level of resuspension that occurred.
Okay, great. Anybody else have a question? Any of the presenters have a question? I don't see any hands raised. Okay, thank you so much, Ashley. That was terrific. So I think we will now move on to our final presenter. Last but not least is Beryl Khan from the Graduate Center of CUNY. And her project is Effects of Coastal Acidification on Jamaica Bay Salt Marsh Nutrient Cycling. And her supervisor is Chester Zarnock. Okay.
Hi, everyone. Thank you so much for that introduction, Helena. Thanks so much for being here this afternoon. My name is Beryl Khan, and I'm about to start my second year of my PhD program at the Grad Center. So I'm hoping that this work from this summer will go towards my dissertation project, and I'm really excited to share with you all what I've been working on so far and where I have yet to go with it. So a quick overview of where I'm going to be taking you all today. I'm going to give some background on my project, my research questions, and the sort of guiding questions behind those. I'll go into my methods that I've done so far, and then show you guys some preliminary results and discuss those with you. And last, I'm going to show what I still have yet to do and what I predict to see from those remaining experiments. So we've heard several different presenters so far talking about nitrogen loading, since it is a huge problem in the Hudson River estuary. As we're all aware, it leads to severe ecosystem service losses. So in my particular study system, I'm interested in looking at how nitrogen loading causes the loss of marsh ecosystem services. And as we know from all of our work thus far, that a lot of this anthropogenic nitrogen in the Hudson River estuary system comes from wastewater effluent and from runoff. And when we see a large end loading event, we see the subsequent events of destabilization of the salt marshes in the area and their subsequent erosion or bank collapse. So this is actually a photo here of a bank collapse in action, this sort of degradation of the marsh edge due to this end loading event. And we also see a subsequent side effects of nitrogen loading in the form of algal blooms, both macro and microalgae, and subsequent hypoxia in the waterways as well. What we also see is a lesser known side effect of nitrogen loading known as coastal acidification. So when we think of acidification events in marine systems, we usually think of ocean acidification, but this is the big distinction between coastal and ocean acidification is that in an ocean acidification event, it's an atmospheric deposition of carbon dioxide into the system. So it's an external percolation of CO2 into the system to lower the pH. Whereas coastal acidification is actually the opposite process, where an internal release of carbon dioxide will lower the pH. And this happens because with an excessive input of nitrogen into the system, algae blooms with their diel respiration in combination with elevated decomposition rates lowers the pH of the system internally. And what we see with these events, what's been documented so far, is that this has both seasonal and diel changes in the pH as documented by Wallace et al. These have been noted to be biologically significant in terms of impacting marine life that experience these. So within the Hudson River estuary, the particular algal blooms that we see pretty prevalently are in the form of macroalgae or ulva. And this particular group of algae tends to cluster around the salt marsh islands of Jamaica Bay. So Wallace and Gobler back in 2015 did a survey to see how much percent cover of ulva was actually in the system. So they did this in 2 different seasons of the year. So in figure A up on top, they looked at it during the fall, and as you can see, there's relatively middling ulva levels compared to figure B down below, which is the percent cover of almost 100% or at 100% in the spring. And you see, in particular, it's clustered around these salt marsh islands. So when we think about nitrogen loading and marsh destabilization, and this introduction of massive growth of macroalgae in this system, the connection between this macroalgae growth and the loss of marshes could have 2 potential mechanisms that I'd like to explore. One of these is altered nitrogen cycling dynamics through the system, and the other is changes to decomposition dynamics. So what is exactly driving this marsh decomposition or marsh degradation process? So the relationship between ulva and decomposition is a hypothetical effect known as co-metabolism, where if you add large amounts of labile carbon in the form of macroalgae, so such as the ulva, it actually accelerates decomposition of not only the labile carbon stored in the salt marsh, but will also lead to subsequent degradation of more recalcitrant carbon forms. And that there's sort of co-breakdown when you add too much of this fast carbon or easily degradable carbon in the system, it then leads to accelerated breakdown of a harder to break down material too. So in my particular study sites here in Jamaica Bay, my 2 sites are Elders East in the north, which was a fairly recently restored site, and Black Bank, which is an undisturbed, unrestored salt marsh as a comparison. So when I say restored versus unrestored, what does that mean? Well, it's a matter of how much carbon is stored in each particular type of habitat. So in a restored marsh, such as Elders East, so my northerly site, is because it was restored as part of the restoration process, they added a lot of sand and fill content to build up the marsh again. But as a result, it has a lower carbon sequestration pool overall compared with an unrestored or undisturbed marsh, such as Black Bank, which does not have that sand and fill content. So it's got a lot more organic matter contained in the sediments and more sequestered carbon overall. And the importance of this distinction here is that it potentially leads to differences in biogeochemical processes of each different type of habitat. And I'll get into that further on. But to sort of circle these ideas all together in the form of my research questions, I have 2 of them guiding my work. So the first one is, how does the presence of ulva macroalgae impact the decomposition dynamics between these 2 types of marshes, restored and unrestored? And secondly, what the effect is of coastal acidification events on the nutrient cycling and specifically nitrogen cycling processes in these 2 types of habitats. So in terms of what what my overall plan has been and will be, it's a 4-part study thus far. So first of all, estimating how much ulva is actually in these particular sites. Secondly, to calculate the decomposition rates of the 2 different marsh types using 2 different forms of carbon, and I'll go into that in a couple of slides. We have been tracking and will continue to track environmental data in the form of pH out in the field, as well as other environmental parameters. I'll explain in a little bit. And we're actually getting these data in real time from data loggers. And last, I'm going to be doing a core incubation study to attest the effects of acidification on the nutrient cycling dynamics of these systems. So what I've done so far, I've completed my ulva biomass survey. So these are just some photos from the field and from the lab. So here on the far left is an image of one of my transects, my high intertidal zone transect in the Black Bank site. And along these transects, we randomly sampled 1/4 meter quadrats and collected all the ulva within those quadrats, brought them back to the lab, rinsed them off very thoroughly to get off any encrusting organic material. And then we dried them to calculate the dry weight from each of these different zones and sites. So this is just a schematic of my transect design. So we have, and this was the same in both sites, both Black Bank and Elders East, where we have the high intertidal zone marked out on a transect right up against the marsh here. The low intertidal transect, which was right at the low tide line at the water's edge, and then the mid intertidal zone marked out intermediate between those two. And another important distinction I should mention here between the Elders East and the Black Bank sites, so the restored versus unrestored sites, is the width of the beach and the height of each. So the Elders East, the restored site, has a higher elevation than the unrestored Black Bank site. So, and it also had a much broader beach. So the width of the Elders East site was far wider, about 50 meters of width from high to low transects than the Black Bank site, which just had a narrower beach and a little less room between the high intertidal zone and the water's edge. So it was about 30 meters in width. So what I've also done so far is I have installed the litter bags for what's known as the T-bag index. So it's essentially a standardized litter bag experiment that was developed by Questionnaire et al. In 2013. And what it does is it uses standardized litter composition in the form of commercially available tea, which is kind of cool. And that you have these two different types of tea that represent both recalcitrant and labile carbon. So you have rooibos tea or red tea, which is much more of a woody material composed of largely more recalcitrant forms of carbon, in contrast to the green tea, which is largely comprised of more labile forms of carbon. So you have these two types, and you can actually bury them in different treatments. So all of one kind in one site and then combined in another to compare the rates of decomposition and see how these carbon types will impact one another, the degradation of one another. So these have been buried in the site for a little over a month. They have to be in there for 90 days. So they're still there. Hopefully the hurricane didn't wash them away. So these have been deployed, and I will get more into the future directions with that later on. But just to show you what the treatment situation looked like in each of the sites, the way that they were deployed, is that in order to test for this co-metabolism effect, I would bury the different tea bags in 3 different treatment levels. So green tea only in one treatment to see this labile carbon only in a treatment, a red tea only treatment to test this recalcitrant carbon only setup, and then a combination of the two, so both labile and recalcitrant, both red and green, to see if there's this co-metabolism effect that's occurring, if the labile material accelerated by the ulva, in fact, degrades the recalcitrant carbon more rapidly. So that's how they're set up. This is not to scale. There are more replicates than that in the sites, but this is just to give you a sense of how they're arranged in each of my sites. I have also deployed data loggers in each area as well. So we have a salinity logger, a dissolved oxygen logger, a pH meter, and a light penetration meter. So all of these are collecting data in real time. So here I am on the left priming my pH meter in the Elders East site. And we've been able to pull off the preliminary data from these loggers. And they're still out there calculating data as we speak, hopefully, presumably. So in terms of what I found thus far, so the results from my ulva survey, and just to orient you a little bit with this figure here. So we have my 2 sites, Black Bank here on the left and Elders East here on the right. And then the x-axis, we have all 3 of my transects, or the marsh zones spelled out here. So the low, mid, and high intertidal zones. And what we see is that the patterns of this ulva distribution relate strongly to elevation. So as I mentioned earlier, Black Bank has an elevation gradient at sea level of around 0 meters. It's basically flat. So we see much heavier proliferation of ulva in the low and mid intertidal zones compared to the Elders East site, which has a difference in height of about 1 meter. So there's a lot more exposed area in the mid intertidal zone. So we see almost no ulva there in the mid because it's just too much exposure during the tides. So we see these patterns pretty significantly linked to the elevation dynamics in these 2 different sites. I also compared the results from my survey with two previous ones done in Jamaica Bay. So one that was conducted in 1995 from the DEP, and then actually a fellow student at the grad center for her dissertation work used those results and compared them to hers in 2012. So I then compared my results to the 1995 and the 2012 data. And what we can see is that both my results and Lamb's results in 2012 both agreed that there was significantly less ulva biomass overall relative to the numbers from 1995. And this does correspond to general trends that nitrogen as a whole has been declining in the sites. But because these are slightly different sites, Black Bank is actually very near Big Egg, but it gives a nice snapshot of the ulva concentration over time. I also have some numbers from the first 2 weeks of data for my pH loggers. And what we can see with the results here is a very strong diel fluctuation in pH in both sites consistently for this 2-week snapshot here in this figure. And what we see is a mean diel range for Black Bank is a pH swing of about 1.4 pH units. And that in Elders East, it's a little higher, it's around 1.5 pH units. And what we can see with each valley and peak here representing a 24-hour period, that this corresponds significantly with the time of the day. So to show you a better illustration of that, I want to zoom in to a day I picked randomly, happened to be August 4th. So we can zoom into that data and look at an hour by hour plot of this different pH data and how it changes throughout the course of a day. And what we see with this snapshot from August 4th is that the lowest pH units are right before sunrise, which makes sense since at night it's not predominantly respiration that's occurring in the system. So we see a correspondingly low pH reflecting that. And the pH starts to climb a little bit before sunrise and then starts to drop off again around 6 p.m. And continues to decline after sunset until dawn the following day. Now, in this particular snapshot, we see a slightly higher difference in pH between Black Bank than Elders East. In this particular example, could be related to the high biomass of ulva that grows in that low and mid intertidal zone in Black Bank. I also wanted to show you the correlation between pH and dissolved oxygen that we have so far in the site. That there's a very significant correlation between these two factors. And the readings correspond pretty visibly in terms of times of night or day of recording. And what we see here, so we have our black points representing those recorded at night, and our light blue points corresponding to those recorded during the day. And what makes sense is that we see that the lowest readings of DO occur at night when it's respiration dominated, utilizing O2 from the system, while we see super saturated readings of DO during the day. And this is probably because our logger is situated right in a very large bed of ulva. So we see this super saturation of oxygen at the peak of photosynthetic activity in the site. So in terms of what I still have yet to do, my next step is to conduct my core incubation. So this is the nutrient cycling study. So what I'm going to do for that is I'm going to collect intact sediment cores from the sites. This is an image of a core study we did 2 weeks ago, so mine will not have mussels in it. This is just to show you what that sort of setup looks like without mussels. And these will be conducted under dark conditions, so no photosynthesis, and it'll be a continuous flow incubation. So mine will be 2 treatments, an acidified and an ambient treatment. So the acidified water will be about 1 pH unit lower than the ambient one. And this is due to the readings that we've seen so far in the site from the pH logger data. And we'll maintain this lower pH by bubbling in CO2 mixed with air. And next, so once those cores are incubating, we're then going to compare water samples from the inflows and outflows of these cores and get the nutrient and gas flux information and compare them between the in and outflows. What I hypothesized to see here is that under ambient conditions, so non-acidified conditions, what we'll see is the dominant process in both types of marshes, so restored and unrestored, that nitrogen removal will be the dominant process there. Similarly, I anticipate that in the restored site, restored sites will also be dominated by nitrogen removal. However, in the unrestored site, so the site that has the higher carbon pool, based on biogeochemical dynamics that rely on carbon, I expect to see higher quantities of nitrogen recycling rather than removal in that particular site. Next, I have to reclaim my tea bags and actually calculate the decomposition rates using the formulas set down by the tea bag index protocols from Questionnaire et al. And I'm actually using ones that were adapted for salt marshes specifically by Mueller et al. From 2018. So I had to dig up these tea bags from the site. My pull date is officially October 3rd, because that marks the end of the 90-day period that they've been decomposing in the site. So I'm going to bring them back to the lab. I'm going to dry them and reweigh them. And based on that mass loss from before and after, I'm going to calculate the decomposition rate using this equation and others down here at the bottom. So I won't get too much into the weeds of that, but this allows you to calculate the decomposition between the different types of treatments. So whether there is a co-metabolism effect. And what I hypothesized to see here is that the ulva will in fact accelerate decomposition rates, but it will be more extreme in the restored marsh than the unrestored marsh. And once again, that has to do with the carbon level in each. That in the restored site, there is less carbon sequestered overall. It takes longer to accumulate there, and hence it will decompose faster with a co-metabolism effect. So we'll see the green tea accelerating decomposition of the red tea due to the presence of this ulva accelerating decomposition overall. So thank you so much to the Hudson River Foundation and to the Polgar Committee. Thank you so much also to my advisor, Chester, and all the people who helped us out in the field as well. There are a lot of names, so just a general thank you to everybody, and I'm happy to take any questions that you have.
Great, that was really excellent work, and I love the tea bag study. So do we have any questions for Beryl? Hold on just one second. From Judy Weiss, wouldn't you learn more if, in addition to the tea bags, you used litter bags with litter from Spartina and compared them with something that actually lives in the marshes?
So that has been done where they've used actual marsh litter, and it does actually work as well. This is more just sort of a proof of concept for the tea bag study in general. And what I also plan to do, since there have been other studies that have done this, that might come along later. This is more just to kind of test for it superficially, and then that might be something I'll consider later on to, first see if this is actually a phenomenon that's occurring, this co-metabolism effect. And if I see it with the tea bags, I'll see if it's worth doing with the actual litter bags going forward.
Good. From Mark Boughton, how much of the salt marsh loss in Jamaica Bay would you attribute to eutrophication as opposed to sea level rise?
So it's tricky to tease those apart, and we certainly see the results of both. But what we have seen as corresponding with levels of nitrogen loading that's been in the system since around the 1950s, that we've seen marsh loss even then. So a lot of these marsh islands have been degrading for decades, even before they started recording a lot of sea level rise activity. So it's sort of a longer trend almost of this nitrogen. But yes, absolutely, that sea level rise does contribute to the degradation of marsh banks as well.
So from Allison Fitzgerald, we've got excellent job. The field design is great. Wondering if there was a difference in the sites with respect to location. For example, was one site closer to urban areas, CSOs, docks, etc., than the other?
So that's a great question, and they're both fairly isolated. The Black Bank site is, it's on the side of the bay that has the wildlife refuge, so it's pretty undeveloped over where it is. And then the Elders East site is an island, so it's pretty much on its own. So they're both fairly far from, I don't believe there's any CSOs in the immediate vicinity of either one. However, I'd say they're probably about equidistant from an outfall that's on the beach of the Cross Bay Bridge, I believe, has a pipe. So they're both about equidistant from that. So yeah.
So from Gustav Kahn, That was a wonderful presentation, very engaging, great work. And from Ray Kepner, excellent presentation. You should present that at the Northeast Algal Symposium in the spring.
Thank you.
Okay, any other questions? Let me see if our presenters have any questions. I don't see any. So great job, Beryl. That was terrific. And I want to say great job to all of our fellows today. Really interesting work that you did this summer. And before we end, I would like to give special thanks to Dave Yazzo and Sarah Fernald for all of the work that they do on this program. And I always want to thank all of the advisors because we really couldn't have this program without your help. So thank you very much. And if you would like to know any more about the Polgar program or about any of the other work that is done by the foundation, please visit our website at hudsonriver.org. So with that, I think that we'll end the day, and I think it was a great day. So I hope that you have a good rest of the day this afternoon. Take care, everyone. Bye-bye.
Bye.
Hi, thanks everybody.
Bye. Thank you so much.